So, good morning. You are here, hopefully, for medical device epidemiology. Seeing some head nods, great. One last reminder, we only call on people in the back. So if you want to have a greater chance of not being called on, move closer to the front. That's all I can offer. We are going to talk today about a few different things. I did remind you that coffee is downstairs, and that is because we start with all of the regulation and methodologies and all of that for medical devices. You're stuck with me for that point. So if you fall asleep, we'll just, yell real loud at the end of that time because you don't want to miss Dr. Christina Mack and Dr. Mackenzie Herzog for their amazing updates on what's going on in the medical device epidemiology world. They talk about wearables, they talk about diagnostics. It is amazing. You'll love that part. It'll be nice after the regulations, I promise. After that, we have David Maislin, who is going to wow us with all of the recent advances in medical device real-world evidence. Nicely, he's a statistician, so he does ground us. He may even have a couple of equations on his slides. I can't guarantee, though, because he's an applied statistician, which means he's willing to talk to epidemiologists. After that, we're going to have our panel just after the break to lure everyone back. Bring all of your questions. Feel free to write them furiously through all of these first three talks. And then we will talk about the future of medical device epidemiology. Paul Coplin is going to share with us some things that have been happening through various networks around the world and really just talk to the latest and greatest of how real world evidence is really driving the science around medical devices in general. And then at the end, Dr. Herzog is going to wrap us up for the day. We'll be finishing up around 12:30, so this is a 4-hour course. We do encourage standing up, moving around, that type of thing, whenever you need to as well. And as I promised, we're going to start with an overview of the regulations and methods in medical device epidemiology. Now, as a disclosure up front, I am a uniquely biased individual. In the past year, I have worked in all three sectors that are part of ISPE. I have worked in government, I have worked in academia, and I have worked in the industry service provider area as well. And so I do feel the need to let you know that all of these statements are mine. No one else has looked at this, and this is definitely not representative of any government thereof. So we'll talk a little bit about study design considerations. We'll talk a little bit about different regulations around the world. But to get us started and to remind you that this is interactive and that we like questions, we're going to start with a game. So our game is this a medical device? Are you ready? This is a hip implant. Is this a medical device? All right, generally yes. Excellent, excellent. We're all on the same page. Perfect. This is a syringe and needle. Is this a medical device? Excellent. This is great. Everybody already knows all this. We can just chit-chat for the 4 hours. All right. So this is a pre-filled syringe. Is this a medical device? This is a drug. So syringe itself, device, but if it comes pre-filled, that's a drug. This is an ultrasound. Is an ultrasound a medical device? Ooh. Yeah, we'll go with that. It's going to be a diagnostic. It's going to be sometimes a therapeutic, but definitely takes pictures. This is a medical device. Here's my favorite one. This is a toothbrush. You probably even used one today. Is this a medical device? Oh, okay. We're in Halifax. Halifax is in Canada. And in Canada, this is a medical device. In the United States, this is a medical device. But in Europe and most of the rest of the world, there's this weird carve-out for the medical device regulations for cosmetics and toiletries. So in Europe, this is not a medical device. It's a consumer product. It's used ubiquitously, though, so you'd be surprised how many adverse reactions there are due to abrasions or other kind of skin considerations from toothbrushes. Be careful when you brush your teeth. This is an Apple Watch. Is an Apple Watch a medical device? Oh. Now everybody's not even looking at me. An Apple Watch is a medical device. So it's not like the watch itself. The telling of time doesn't make it a medical device. But an Apple Watch has on it a heart rate monitor. Some of them have EKGs. Mine has told me that I am breathing too rapidly or not breathing deeply enough. It's a medical device. And it has been cleared through multiple regulatory agencies around the world. Now, the medical device industry itself is about 500 billion per year, medical or industry in general. It is expected to grow 50% or more in the next decade. So it is a very rapidly growing industry. And in the next 10 years, this increase is anticipated because around the world, populations are aging. Around the world, people have a lot of chronic conditions, and an aging population with chronic conditions needs more medical care. There's also increasingly an emphasis on having early diagnosis and early treatment. And when you need an early diagnosis, you tend to go to a medical device, a diagnostic, in order to get that information. And often, early treatments are also medical devices. Then there's more surgeries, and those surgeries may have a more technical component to them. And really, there's technological advances that are happening in all industries and all fields, and most certainly in the medical field, and that falls under medical devices. So you are here today, which means you are ahead of the game. You're learning all the ropes. So voila, there's plenty to be done, and we're so glad you're learning about it. The top therapeutic areas are in vitro diagnostics, diagnostic imaging, but then also cardiovascular and orthopedics as well. The regulation of medical devices is still relatively new. The first country around the world that had medical device regulation was the United States, and that was in 1976. So it's not even 50 years old anywhere in the world. And most countries now do have a regulatory section that is specifically for medical devices. And when there's not a specific section for devices, then these medical products that fall in this realm are regulated as if they were drugs anyway. So with pharmacoepi, you're usually pretty used to the drug kind of milieu. We're just going to add on to that the device one now. And I've talked about it a little bit, but you may even want to know, is there a definition? What exactly is a medical device? Well, this is the Australian definition. I like it because it's nice and laid out pretty. A medical device often is in a human being and used for a human being, though as a special caveat in the United States, it also includes animals. A medical device can be used to diagnose, to prevent, to monitor, to treat, or to alleviate a disease, to work with an injury or handicap, to replace some sort of anatomical or physiological consideration or process. It can also be used to control for conception. In addition, a medical device is something that achieves its intended action by some means other than a pharmacological, immunological, or a metabolic means. So basically, a medical device is something that's used in a person for some sort of medical purpose that is not a drug. So the medical device regulation holds everything, and there is a carve out for drugs. So if it's not a drug and you're using it for a person, for a medical purpose, it's a device. There's a lot of nuances to this definition, though. I like the Australian one. It's nice and pretty and laid out. But in the United States, there's that non-human clause. In the EU, there's that toiletry and cosmetic clause. And in some jurisdictions, there's a lot more that's required in order to be on the market as a medical device than in other places. Now, there are several types of medical devices. They can be therapeutic in nature. It can be a drug delivery device. And so often, if it is a drug delivery device, if it is separate from the drug, like that syringe that was with the needle but no drug, that's a device. But if it is together with the drug and its packaging, then it may not be considered a device. It may be considered a drug. But that drug delivery component, a lot of devices fall into that. The device can be diagnostic, radiologic, or monitoring in nature. And these days, there's a lot of devices that are software or apps or have some sort of component of that. And it's a medical device and needs to fall under the medical device regulation if that product is fitting into one of these types by itself, or if there's a component of the product that fits into one of these different considerations. So even if you're studying a medication, and even if you know that you want to study drugs for your entire career, knowing a little bit about devices is really helpful because it will give you some nuance around what it is that you are most interested in for your career. So again, we're really glad you're here today. The regulation systems are a little bit different for devices than drugs. In some jurisdictions, there is one regulator that regulates drugs and biologics and devices. In other jurisdictions, in this, interesting, sorry about that. In this particular situation, it allows for the different people who are doing the reviews for the drugs and the reviews for the biologics and the reviews for the devices to talk to each other a lot. And so when we have these combination products, you can have specialists in each area speaking to each other. In other jurisdictions, there's a government body that is over the regulation, but then the specifics for the regulation for devices fall to other entities. This is what happens in the EU. And there, each country has its own group that provides regulatory oversight. And then they empower other entities, several of them per country in some instances, to conduct those reviews. So you have, hundreds of entities throughout Europe that may be the group that reviews and approves that device to be on the market. They all roll up into the same legislation. So they have kind of a general framework that they all use. And this allows for many more products to be reviewed quickly. So there's some pluses and minuses to both. I won't bore you further on it. It's really exciting. One is much more like drugs. One is much more like consumer products. But if you want to know more, that's a great question for a panel. So the standards. For drugs, the standard is that for a new drug to be on the market, it needs to be safe and efficacious. That is in perfect conditions in a clinical trial. You can show multiple times that with the drug versus without the drug, you see a difference in how those people have their outcomes. Effectiveness is the bar in the United States for devices. And that is within a clinical context, you see a clinically meaningful difference in the outcome with and without the device. In Europe, the standard is clinical performance. It's very, very similar to effectiveness, but there are sometimes some nuanced differences in just how you need to design your study. And that means that within a clinical context, often compared to standard of care, if there's a comparison at all, that you can show that the device does indeed work in the way it's intended, that it achieves its clinical purpose. There are also some nuances in the companies themselves. Device companies, by and large, are smaller. One of our presenters today is from one of the largest medical device companies in the world. But that is a little bit different. Many device companies have one product or two products, over 70% of them, in fact. And so they are small companies. And many devices, they focus on that product, and that product is a physical product in nature. And so they will make tiny modifications to that product over time. And those iterative modifications, because it is a mechanical product, because a lot of the testing can be done at the bench, those modifications can occur frequently. And so because of this, the product life cycle is shorter. Historically, The drug life cycle was about 17 years. The drug would be on the market for about 17 years. Historically, for devices, they were on the market about 17 months. Right now, because of AI, because of all of the exciting things that are happening in the technology space, in some areas, it is a long time if a device is on the market before the next iteration for about 3 months. So huge, rapid cycles here. Nonetheless, getting to the market has a similar feel to it. For drugs, they do indeed start at the bench, move into a clinical trial. All that information from the non-clinical and clinical studies goes into a marketing application that's reviewed by a regulator. After that review, there is marketing authorization, and then the drug moves into a space of post-marketing surveillance, risk management, and safety and effectiveness. For a device, those same steps do occur. They occur a little bit closer together, and they have a little bit of nuance to them. There will indeed be some sort of lab or bench or non-clinical testing and development that occurs. There may or may not be clinical studies that happen pre-market. Most products do come to the market for devices without any clinical study. If there is a clinical study, it looks a bit different, and I'll share that in just a moment. Most devices do need to have a marketing application to them, but in many cases, that device does not have to stand on its own. It can reference other material from previous iterations or other similar devices. And so that marketing application might be, yes?
Sorry to interrupt you. When you say no marketing authorization, is that the United States or does that include Europe?
I've been covering both the United States and Europe and will continue to do so throughout this presentation. And it's not because only the United States and Europe are important. It's because these two frameworks tend to map around the world. In sub-Saharan Africa, about half the countries map to the way that the US does it. About half the countries map to the way that Europe does it. India looks much more like the United States. Canada has this interesting hybrid approach. But these two frameworks tend to be at a macro level, generally the two ways that it happens. And I'm looking at David and Paul, and neither of them is like, oh, this is terrible. So this is generally how it goes. As you may have noticed, a lot of nuance here. And so if you're looking to focus on South Africa, we can have a separate conversation about that later. Sorry, Paul, we are recording, so if you could grab a microphone real quick.
Good morning. Good morning, everyone. Good morning. Okay. Just, I think the big fact has been that if any country adopts a different approach to medical device approvals, they tend to be the laggard in getting the device on the market. And so the technology starts to lag in that country. So we see a lot of governments really looking to how can they streamline their approval process to be more harmonized. Because if they impose a new requirement, then what happens is the product gets approved elsewhere, and then they have to do that requirement for that country. And then to get the technology gets there 2 or 3 years later, so they're lagging on the technology.
The point is why we're here, because Europe was regulation light from devices perspective, and in the last year or two, things got really hotter. And now, like for example, in the UK in January, they also introduced very, very more strict regulations. So I just was wondering whether you're covering the lighter or the new regulations.
The new regulations have been written for a while, and we've had them incorporated in the course for a while. So you're welcome to go back to previous years to see how we discussed them in previous years as well. But we do indeed cover both. So where were we? We were at the marketing application for devices, which does indeed happen for most devices. And for those that are higher risk, it does look very similar to drugs. For others, there's a bit more nuance to it, a little bit lighter. Nonetheless, devices also move into, after authorization, this post-marketing surveillance, risk management, safety effectiveness need. All of this nuance is really based on the classification. On the slide here, it's listed kind of the higher ones on the slide are the lower risk devices. And then as you move down the slide, those tend to be a bit more high risk. And so you do need more information to support that application as the risk of that device increases. And so I listed here on the slide the EU, the US, Australia, and China, just to give you a little bit of a flavor. And so in the US and EU for that class 1 device, mostly you just need to register, hey, we're going to put this new thing on the market. And that makes a lot of sense for like a wheelchair that's not powered, that you just say, hey, we have this new wheelchair, we're going to start marketing it. For most of those class 2, those kind of mid-level devices, most of them need to show, in addition to the registration, as part of their application, they need to show that you can sterilize it, that it can be cleaned to the level needed in order to use that product. Though there are some class 2 devices that require other special controls, and some of those are clinical studies. And so a device that requires, a device that helps to stimulate bone growth may need a clinical study before it can go on the market. A device that is a coil, so it's a mechanical piece itself that is put into the brain and stays there, might need a clinical study before it goes onto the market. But both of those are that mid-level risk for devices. But those that we don't know as much about, where they're brand new technology or where they're technology that can have a lot of variation for the experience for patients, we still want to have clinical studies before those go onto the market. So those class 3 critical devices where there's just not as much information on benefits and risks, or where the benefits and risks are likely to have a lot of nuance for that specific product, then we need to have that information pre-market, and then generally follow up as well post-market. So those lower devices, maybe just a validated design change for an AI, that we made this tweak, it still has the same sensitivity and specificity. It might be that we need to show that the engineering standards at the bench, there is indeed a checkoff, all of those types of things. So this is just examples of the types of information that might need to be shared pre-market. Those clinical studies, when they do happen, look different for drugs and for devices. For drugs, 1,000 people in a clinical trial pre-market, in many cases, is kind of small. Many thousands is very normal. In devices, a 400-person study pre-market is a lot of people. Often 50, 100, 200 is where you're going to go. And so it's very much to understand if there is a reasonable assurance of safety and effectiveness pre-market so that that new technology can get out to patients. In addition, blinding or masking is very much the norm in drug studies, and the comparator is often a placebo. But for devices, especially a procedure, it's often considered unethical to cut somebody open and to not help them, specifically that individual. And so it's very, very rare to have a placebo or a sham procedure. And it's very rare that that individual would not know what's happening to them. So there's some variation there. Also, the risk while taking a medication is often very well known, and it's often relatively stable throughout the time the individual is taking that medication. For a device, the risk that they are experiencing varies across the time that that study is ongoing. The surgery itself has a different risk profile than the longevity of the implant and the potential risks that the person's having while they are experiencing 1 year or 2 years with an implant. Often, if there is a major adverse event, then a repeat procedure is going to be needed. And so there's additional risk that could happen at that time. The provider themselves adds to the risk profile because providers are more comfortable with specific types of surgeries. And So in those instances, that particular choice of treatment or implant, their comfort level with it will play into the risk profile. And then for many devices, sometimes there's an electrical component that's turned on, and sometimes there's an electrical component that's turned off. When the electrical component is on, there's a higher risk generally than when it's off. And then each person has a different anatomy, say for their blood vessels or whatever it may be. And so if you have more torsion in your veins, if there's more curvature and more twist to them, that's going to put you at a different risk than someone else. And then again, there's other considerations for drugs. The dosing, the adherence to the drug can really play into what you see within the clinical study. And then for devices, the learning curve for the individual who is using the device, as well as the human factors, the interplay between the person using the device and any screen they may see, any component of that device, any piece of the use, and how that plays into human factors also matters a lot. Nonetheless, real world evidence and kind of what we do in epidemiology is the norm in medical device clinical studies and has been for quite a while. It's expected pre-market, it is expected post-market, it is expected early and late in both situations, and really throughout the total product life cycle. Epidemiologically, you're going to see assessments of use, of safety, of effectiveness within typical clinical practice. You're going to see surveillance for unanticipated events. You're going to see long-term evaluations frequently for an implant that's going to stay in the body for 20 years. Long-term really means long-term. A 10 or 15 or 20-year study is not outside the norm. And then also subgroup characterization is a big deal. Many devices look and act differently in men and in women. And so there's consideration there that happens from the get-go. The conduct of procedures themselves and the learning curve is really important there as well. When we start to think through the additional real-world evidence assessments that are needed, there's a need for active surveillance to really think through what we are likely to be seeing and to be able to characterize that well once the device is on the market, because we've really only had a couple of hundred people at most who have used it pre-market. And so knowing quickly if there is an issue and what that might look like is really important. And then there's a lot of changes in the medical system, and as we've already discussed, within the technology for medical devices. And so understanding how the disease and the condition prevalence and the validation of the biomarkers plays into all of that is really important. And then because of that iteration in the post-market, we learn what's most needed for the next iteration. So we use that cycle as well as additional assessments in the epidemiological space, and really to understand what's happened in the past, what can happen in the future, and historical and concurrent controls that are from the real world as an external control are much, much more used in the medical device space than in the drug space. So nonetheless, I'm going to say a whole bunch of words really quickly that you already know. You're still going to see confounding by indication. You're still going to see channeling bias. You're still going to see healthy initiators, healthy adherers, healthy user effects in general. You're still going to see heterogeneity of treatment effects, and you're still going to need to validate your outcomes. So basically, all the things you've learned in school, you got to apply in devices. The only things you really need to learn are how to evaluate provider characteristics and how to evaluate a learning curve. Provider characteristics, the quick and dirty approach is to look at surgeon volume or to look at training or to look at years of schooling. There's often just a need to understand when a surgeon is doing a procedure, is there a break point in how many procedures they need to do per year in order to remain competent for that procedure. In this particular evaluation of total hip replacements, there's a pretty clear break that if a person is doing at least one a month, that is 13 procedures or more, then the unplanned readmission rate is a whole lot lower than if they're doing less than one per month. For a learning curve, anytime someone is learning to do something new, it takes some time for them to be more comfortable with it. And so as a surgeon is learning to do something new, as a nurse is learning to do something new, they are also going to have to train on it. In this particular situation for total knee replacements, this group of surgeons was switching from a laparoscopic to a robotic procedure. They took their last 10 cases laparoscopic, mapped out the timing, how long it took them to do it, and then they counted. Well, here we go. As they're learning the robotic surgery, yeah, it takes them more time. And in different trajectories, as you can see in the panel on the upper right, in different timing, at different stages of learning, they all increased and decreased back to being able to do the robotic surgery at about the same amount of time or less time. But each of them had to go on their own journey. And so that learning curve and really being able to evaluate that is really important. And it means that as we're considering epidemiological studies, we may not want to take everybody's first 10 of the new procedure because they may not have learned all the different nuances for this new approach at that time. So there's other considerations for real-world data. We talk a lot about data sources throughout this conference. You'll probably hear a fair bit of that over the next 3 days. We'll talk a lot about claims and electronic healthcare records. And that's bread and butter for drugs. In the device area, yeah, an electronic medical record will often work. But we may also need a lot more information on a procedure itself or on the particular AI that was used in order to evaluate that. And so often we might need to link the long-term information of claims to a more in-depth approach, which might be an EHR and might be a registry in order to get that rich exposure data. There is a unique device identifier, UDI, and so that looks somewhat similar as a coding schema to the drug codes. However, the unique device identifier, that UDI, is not often available within claims or within EHR. And so there's still a need to do quite a bit of natural language processing or other identification measures through the clinical notes in order to find the specific exposure of a device. Nonetheless, the outcomes do look very similar to drugs. We're going to be looking for procedures, for diagnoses, and we will need to look many years out. And so in the device world, they have done quite a bit of work to be able to not identify a patient, but still be able to link their data over time. And so this patient privacy protecting data linkages, which is actually a symposium later this week, that's a big deal for devices and has been used successfully to look for 10 years or 20 years of outcome data. But the procedures themselves, like a repeat procedure, you're going to see that in claims if you're looking long enough. And so that can be very helpful. For the patient population themselves, a device study often will look at everyone who is using that device. They won't necessarily pull down to this specific indicated use. That can be in part because many devices have an indication that is very broad, and so many populations may use that device. And it can also be because the post-market surveillance, where we would have this real-world data, is intended to capture as wide a net as possible to better understand adverse events. And indeed, the details of the prescription itself, the rationale for why this particular device is being used, and the specifics of the devices themselves doesn't necessarily fall under a separate rubric as it would with drugs. In the United States, there's often even a separate categorization, separate set of the data that you have to purchase separately for the drugs. You don't have to do that for the devices. It's all going to be kind of in that EHR capture. The device timing is really playing out in many different factors. We talked a lot about kind of the technology shifts and the need for long-term data And so we're looking now to see that we're more likely to have a shift in the clinical data collection to post-market, so that we can get these technologies to people quickly, so that we can really get that reasonable assurance, and then move to a post-market larger data capture. And it really allows us to have a better use of real-world evidence and real-world data itself. Nonetheless, there are some traditional bread and butter post-marketing studies that we see for medical devices. In the US, those are called post-approval studies. So for those who are playing the game at home, a pass in devices is a US thing, not a Europe thing. Seeing nods. I know everybody's like, okay, sure. And then in Europe, a post-market clinical follow-up, a PMCF, is the more typical approach. Both of these may be looking at outstanding questions. Both of these may be looking at longer-term follow-up. Both of these may be looking at additional data for subgroups, as well as learning curves and provider effects. In the United States, those past studies looked at the safety of approved drugs, and then you also have other kind of risk mitigation style efforts that are somewhat similar to drugs. They show up in annual reports. They show up in for-cause post-market surveillance studies, which are not PSS. They are called 522s in the United States. And that is just, it's the regulatory jargon. And so it's just kind of how it falls under the efforts there. The safety signals themselves and the evaluation of the safety signals, there is a passive surveillance system. The medical device reporting system is called, in the United States, is called the Manufacturer and User Facility Device Experience, MAUD. Worst acronym ever. So post-approval studies can only be required for those highest risk devices. There's very specific needs for them. It looks a lot like PMR and PMC for drugs. For 522s, they can only be asked for those mid-level risk and high-level risk devices. And they are have to have a serious adverse health event that needs to be used in a pediatric population, or it needs to be implanted in the body for more than a year, or it needs to be a life-supporting device. So there's very, very small set of criteria that can be used for a 522, but a 522 can be for cause at any point in the device effort itself. And they are also set as a specific duration. 522 can only be 3 years long unless it's pediatric. For Europe, the post-market clinical follow-up is used to evaluate safety of an approved product or to get more additional data on it. The risk mitigation is covered under 2 different aspects: the clinical evaluation report and the periodic safety updating report. The CER has been around for quite a while. Periodic safety updating reporting is new under the new regulations that have been around for about 3 years now. And then the post-market surveillance, this is PMS, is in Europe. That post-market clinical follow-up is required if there are residual risks or there's lack of clarity on the long-term performance. And these have historically been kind of black box in nature. But under the new regulations, there is a more transparent process. There are more people who are involved. And these are becoming much more available for folks to look at. Real-world evidence has been used a lot in the medical device world. And there are 90 different examples that the FDA put together a couple of years ago. They are vast in nature. There's pre-market uses, there's post-market uses, there's peri-approval, there's all sorts of things. And some folks today are going to share a lot of those. I wanted to share one because it was for a surgical robotic system, and it was using only the data that were already available in an EHR for a pre-market approval, because it was an extension of the labeling for a robotic surgery device. And so they were looking to expand from general GI kind of consideration to being able to use this particular surgical robot for gynecological surgeries. And so they looked at an EHR and looked at literature. The EHR itself, they evaluated 45 patients that had used, or for which gynecological surgery had been done using this robot. 45 patients. And in the literature, they found 8,000 operations. And so they were able to compare the outcomes in these 45 patients in this EHR to what was seen in the literature. And that was enough to confer pre-market approval. So I wanted to just use this as a closing thought to say it really is a big deal. The techniques that we learned in school as pharmacoepidemiologists are used frequently in the device world. And so I would encourage every single one of you to listen to all that's going on today, ask lots of questions, and if you have interest in this field, jump in. There's a ton to do. It is so much fun, and pharmacoepidemiologists can really advance this field well. So I think that I'm already 2 minutes over time. Are there any thoughts, questions, discussion points, clarifications before we jump into our next speaker, who I promise will not be telling you about regulations? Yes. Hi. Absolutely. The clinical context of how a device is being used is just as important for a device as it is for a drug. And you're right. There is a lot more going on for the device. The information is also in different places and looked at in a different way than it is for drugs. And you have to have different people at the table in order to have all the perspectives together. But it is absolutely critical and just as important. Yes.
One of the reasons that the NHRA delaying the regulations that you said for reference employment is because they can't recruit people who have like engineers and IT engineers to know the technicalities of a double chamber or defibrillator and all these complex stuff, robotics and so on. So when we evaluate an individual case in pharmacovigilance or medicine, we can't do the evaluation. We call it a ophthalmologist or a neurologist or whatever. Here, it looks like the expertise of people who are knowing how the device works, almost exclusively are within the company or the people who are advising the company. They're very, very few people.
So I have not worked at a device company myself. I have worked in multiple different places that do evaluate those types of things. For an individual report, I mean, public knowledge, for an individual report at the FDA, the people who are reviewing the individual reports for devices are by and large nurses and biomedical engineers. And that has been my experience pretty frequently across the entirety of the field, is that you have to have both of those sets of expertise. And it is very, very much the nurse who is involved. It's part of what drew me to medical devices because that's my background. So, Christina, we are running over time, and so I'm going to go ahead and hand this over to you.
Just to add to that, think back on, this is a patient advocacy moment of the day, like Mary Beth's chart about the impact of the provider experience, I think is kind of what you're hitting on, right? Like the device can work perfectly, but if you get an individual report about a device, maybe it was the doctor's third time to put it in. I mean, that is relationship without question. So, a couple lessons learned. Yes, it's confusing to figure out. It's hard to figure out if a device works because so much more than a drug, it's dependent on the setting in which it was put in. And a drug, even if your provider is me and I'm not a doctor, I'm still just giving you a pill and you're swallowing it. I can't totally screw that up unless you don't know what to do with it, right? So, second point there is if for all of you taking care of parents and children, ask the doctor how many times they've done the surgery before you let your parents go into the operating room. All right, so Mary Beth was kidding when she said I wasn't going to talk about regulation. So I'm going to kind of walk a little bit through the journey that she just took you on for the first few of my slides and emphasize some of the points she made with a case example. And then I will pass it over to my colleague, Mackenzie Herzog, who will continue that and give us another example. So I'm going to talk about what I think is bringing us toward the future. We just talked about how complicated it is to study medical devices. And so what I want to talk about with you today is what is the new way we can study medical devices? Mary Beth mentioned data sources. She mentioned the power of linking data sources together. So I want to talk more about that in the context of regulations and guidances, and then give you some case examples of that in the context of sports. So here are my disclaimers. I'm a full-time employee of IQVIA. I own stock in J&J and AZ. I did not do any of this research by myself, so I'm powered by an amazing team, most of whose names are listed here on the slide, one of who is sitting in the audience with us today. Oh, sorry. Can you hear me now? Oh, thanks. Okay. So Mary Beth kind of took us through, hopefully everyone in this room can teach this slide. She kind of took us through from a regulatory perspective. She kind of took us through what is required for medical devices in the RTC space. And then when we move out of that space where we've only seen 100 devices in a really narrow population without much follow-up, we're going to move out of that space into the RWE space where we have big patient populations, really diverse sets of doctors, sets of experience. My dad always says 50% of doctors graduated in the bottom half of their class. So you've got doctors that don't completely know what they're doing treating patients. It's not those that enrolled in a clinical trial and do research. So what I'm going to talk about is going to be in that kind of RWE space. And the reason that I think it's really important for us to talk about this in a different way from a medical device space is that medical devices are actually giving us information in a completely different ways than drugs are. So when you use a drug, it's not going to give you information. When you use a device, actually, in most cases, it is giving us information. So Mary Beth talked about product and disease registries, claims, and EHR. Those are powerful kind of foundational clinical existing data sources that we can use. We can link them, or we can use them separately to understand patient health and medical devices. In the medical device space, and to some extent in the drug space, we have other data sources that, particularly if we link them with these foundational clinical pieces, we start to get even more rich information. So laboratory data. When patients go into a lab or use diagnostics, we can get constant streams of those results coming into what I'm going to talk about, which is a kind of linked growing evidence hub. We have mobile connected information, so we can take patient surveys, patient PROs that come directly from a patient's device. And then we have patient-generated data and device-generated data. And this is the kitchen sink. So this is a toothbrush. Okay, just checking to see if you're paying attention. It's not a toothbrush, but it is wearable devices. So it's your ring, it's your watch, it's a wearable device that measures your running, your accelerations, your intensity of activity. Those devices, as well as ultrasound devices, are measuring adherence, they're measuring movement, they're measuring the patient experience. We're hearing directly from the patient through those devices. And we're at a place where we can link that into these foundational clinical EHRs, claims, databases, and have a really, really rich database that can help us with how overwhelming it is to study devices, because we have a lot of questions about the device and we can ask a lot of questions to these big linked evidence hubs, which is why I feel like those are the future. So we talked a lot about all the different kinds of devices. The point I want to make today, or the case I want to make, is that if we bring all those devices in and link them in a meaningful way with patient clinical information, we can ask a lot of the questions that we have over time and continue to get meaningful answers. And so I'll talk about that in the setting of two different examples, one in infectious disease epidemiology using wearables, and then one in sports injury epidemiology. So this is from Mary Beth's talk this morning. I want to make one point about RWE and what regulators think about what I'm about to say. as she said, there has been kind of an increase in global regulation specific to devices across the world. At the same time, we're seeing a growing recognition of regulators saying there's major differences in studying drugs and devices, and we need to kind of come along with the industry and with the data sets that are available to let industry innovate in the way that we want to study devices. And so regulators have decided to put out a lot of guidance documents, not new regs, to help all of us figure out how to use RWE and answer questions about the devices, both for our understanding of patient effectiveness, use, and safety, but also for regulators to use in what they're doing. And so a lot of this started in 2018, 2019. If some of you have been around for that long, we've seen a real sea change from regulators because of the 21st Century Cures Act and also because of the European Medicines Agency's adaptive pathways guidance. We saw regulators say, we have to start to figure out how to use RWE because Congress in the US with the 21st Century Cures Act is making us figure out how to use RWE. So when these acts and guidance documents came out asking regulators to figure this out, regulators started to figure it out. And so these are individual lists of guidance documents in the medical device and medtech space that different regulators have put out for us to figure out how to use this. Now, they're not new regulations and rules. They're guidance documents that tell us how regulators want to consider using RWE. Mary Beth showed us the book that the regulators have figured out how to do it, but it's taken them a little more than 5 years to figure out how to do that. And they're now kind of, really able to say yes to RWE as we move forward. This one is specific to the FDA in the US. And the reason I put this up there is that what they've really asked for us to do is to use RWE to support label expansion. Mary Beth talked a little bit about this, and determine the usability of RWE for expanded indications, new clearances. The reason that I bring this up is because when we talk about linking these data together and creating these evidence hubs, we have to always be thinking about the regulatory considerations that Mary Beth went through, as well as reading the continuous guidance documents that regulators are putting out, kind of shaping the way that they're going to accept it for consideration. And that should also shape the way that we want to accept it, even if we're not resubmitting it to a regulator, but it's kind of helping us figure out how we want to use it and how we want to trust it. I love this quote from the FDA. This is the book that Mary Beth brought up that talks about, by unleashing the power of RWE, we can accelerate medical product development and bring new innovations to patients faster. And in this same meeting, he also said, give us a chance to say no. So what we're seeing from regulators as well as manufacturers is build it, study it, show us your study design plans, give us a chance to say no, because all of these data are out there and it's a watch. And so it's very much in the gray area of medical device versus a consumer product that tells us the time. Be creative with this and then give us a chance to say no, because we've got to get answers to these questions. I'm going to go into, I think without further ado, the case examples. Before I do, one piece I want to talk about is the patient engagement. So Mary Beth made a couple of references to this in her talk, and Mackenzie will as well. We are now more into the what is happening on the side of the patient than, again, we were 10 years ago. So again, when you kind of talk about this seems really complicated, one of the reasons it's complicated is because patients are the ones using the devices. Again, they didn't just swallow a pill and they're done with it. So when we think about evaluating medical devices, we've got to think about what's the patient experience and use and engagement with that device, which means we've got to figure out how to hear from the patient. And that comes through, ironically, through devices, because that's how I think all of us want to tell people about our health is through our phones, through our rings, through our watches, through the way devices are kind of measuring our patient experience. So FDA, again, has done a really nice job of kind of giving guidance on that for us to use and to kind of help us shape how we want to study devices. So I'm going to give a couple of different case examples and then pass it over to Mackenzie. And the key point is in both of these examples, we are asking a lot of different questions from one data source that we've built that links together a lot of information. So the first one's going to be in the setting of COVID. It's a linked real-world data sets that has symptom data from a phone, lab results from diagnostics, vaccine records, wearable devices from Connexon, which is a motion sensor device, contact tracing, operational reports, EHR infection history, so it's built on that EMR foundation, and then sports schedules and stats, which I'll talk a little bit more, but it gives us that denominator information about like how much was a player participating or how much was a patient exercising in this situation. That case example in COVID talks about the use of RWE to understand the effectiveness of diagnostics, and also gives a case examples where we used wearable devices to explain how an infectious disease was transmitted. I'm then going to pass it over to Mackenzie, and she's going to talk about a similarly constructed data hub that we've had for 10 years and are continuing to build. Again, built on that EHR foundation, where we have game statistics, player information, and then 10 other data sources, including wearables, that can tell us about athlete health, and help us to figure out how to reduce injuries and how to understand, again, effectiveness of devices. And I'll pass it to Mackenzie for that portion. So why are we talking about sports when we just spent an hour talking about FDA regulations? That's a good question to lead us into this. One of the reasons is that as the FDA has kind of decided how they're going to figure out how to make decisions on product using RWE and industry, we have also been trying to figure out how do we want to use RWE to make decisions. We've been a little bit of a slower train. In sports, they have always been open to wearables, and they've always been open to athlete-generated or patient-generated health data sources to make decision making. It is a statistics-led group. They've always had cameras counting things that they then decide, is this guy going to go play? Are they going to be a starter? Are we going to pull him back? They've always been measuring at least performance, if not health and wellness, using these devices. And so they're a little bit ahead of the curve in terms of how much they're willing to use these devices to make a decision. They also have a really established focus on health and safety, because athletes, from a business perspective, these organizations need to keep the athletes healthy. It's obviously the right thing to do, but it's also their investment is having those athletes play, which means that they're going to invest a lot in devices and also in studying those devices, and in this case, in constructing these big evidence hubs so that we can ask a lot of questions and get a lot of evidence about how to keep those athletes healthy, safe, and out on the field or out on the court. Which makes sports a really interesting place to look at this, and also makes sports a place that when they've constructed these evidence hub, a place we can learn a lot. So one of the things that happened during COVID, which I'll talk about first, is that the sports organizations had a lot of financial incentive to keep sports going when the rest of the world was closed down. So if you are a sports fan, you probably remember the day the NBA shut down, because it was kind of the precursor actually to the WHO announcing that the world was shooting down. Coincidence or not, but there was a player that went out in the court and then tested positive for COVID. And I think everyone kind of called uncle, and our kids were all home from school the next day. At that moment, the NBA started to think about how were they going to kind of restart their season. And they were going to do that under the construct of daily lab testing. So they tested every single person every day with a sensitive RT-PCR, and started building a 3-year-long longitudinal data set that was linked with those 8 sources I just talked about. NFL did the same, and because Mackenzie and I and our team at IQVIA support both of those organizations, we built those databases right alongside the databases that we've built for years showing their injury data. So this Wall Street Journal article where it says scientists need help against COVID-19, they asked sports, was actually a write-up of, I think, 14 different publications and kind of public health interventions that the sports organizations put forth and then talked to the FDA and the CDC about at every turn during the pandemic, because we were learning so much from these evidence hubs at every moment. So when Omicron came out and they needed to know if vaccines worked, these evidence hubs weirdly were the ones that were able to answer that question, kind of at a surprise to many of us, but because, again, of that investment in building the database. So one of the keys that a lot of us have taken out of that is that we've kind of got to make that investment in these databases when we're not in this emergency pandemic setting, because once you set them up and you bring in all this device information, you really can't answer questions about devices, interventions, products, diagnostics, kind of everything across the board. So as I said, this database had daily for both the NBA and the NFL, and I'm going to mix them as I talk, had a EMR base, and then we were testing everyone every day with sensitive RT-PCR tests. And on many of the days, we were running samples on more than one manufactured product. So we might run it on both a Hologic and a Roche in one day, 2 different RT-PCRs, and give that person a point of care test. So millions of dollars being invested in this, lots of tests being used, and a big commitment to research and helping the FDA and CDC understand transmission and also understand the effectiveness of the diagnostics. So I'm going to give you some results. So in the context of that big database, we kind of had taken a proactive approach in setting it up, and then we're bringing information into it every day, and we're analyzing it and doing a fast kind of agile turn on analytics every day. And we're trying to answer the questions that, as it turns out, everyone else was trying to answer. So in this case, we had connects on device information. Connects on a device about this big. You can set it so that it beeps if you get within 6 feet of another person that has the device, and it actually measures all of your movement. So it actually measures not only kind of who you were near, but also where you go, how fast you're moving, et cetera. And all of that, those terabytes of data went into this database and linked into the EMR, the lab data, and those other 14 data sources. So from that, we had an outbreak. And this was at the time where the CDC recommendation was 15 consecutive minutes within 6 feet was the way that COVID was transmitted. And because we had all of that information in this database when we had an outbreak of 20 cases, we were able to prove that it actually could be transmitted in 4 minutes, which I think, of course, all of us are like, of course it can. But at that time, it actually changed the CDC guidance. This was an MMWR as well as a public health kind of push promotion from the CDC for changing that guidance from cumulative to consecutive, and also talking a lot more about it's really not just time and distance, it's masking, it's ventilation, it's the not riding in cars for 3 minutes with someone that has COVID. So this database was able to prove for the first time how COVID could get transmitted and how long and in what context. Similarly, because we had all of those diagnostic products in, and this is what's going to kind of harken back to that post-approval regulatory space that Mary Beth talked about, because we had all of these diagnostics being used daily on the athletes, both in the NBA and the NFL, we had a closed ecosystem of about 12,000 people that were getting tested daily with head-to-head comparisons of the diagnostics. And then we had daily follow-up on them to find out if they really did have COVID or if they did not have COVID, both clinical follow-up and also additional test follow-up. So we were looking at symptoms as well as test results. So because we had this, we had 11,668 players and staff receiving over 630 to 100,000 tests over a short 4-month period of time. And at the same time, the FDA was really interested in if these diagnostics approved under emergency use worked to detect COVID. And we all know from using the antigen test early in the pandemic that some of them didn't work. You needed to be a lot sicker for an antigen test to turn positive. So we were looking at that, and then we were comparing that with the 4 different brands of RT-PCR tests. So if you look here, what you can see is that between Roche, Hologic, and Thermo Fisher, which were the tests that we were using, we had a positive predictive value range of 73 to 82%. When we did a confirmatory test, it brought it up to 95 to 100%. These tests were extremely sensitive in picking up the virus very early in infection. And what was most interesting about this, particularly to the FDA, is that some of those products were actually only approved for symptomatic detection. So when we did the analysis for this, what we found is that even the products that were only approved for symptomatic detection were incredibly effective, and in some cases more effective for picking up asymptomatic cases versus symptomatic. So we saw really high sensitivity in PPV. When we looked at the antigen point of care, it did not look that way. We saw a sensitivity of 57.7%, and we were able to see, kind of because it was same-day diagnostics with that clinical information merged into it, we could see how long it took these to detect a case, and also, again, could kind of change the protocols and advise to the CDC how to use the many antigen point of care tests that they had purchased and how to kind of instruct the American public to use those as well. And again, just from a public health perspective, one of the things I think that these evidence hubs, as we all kind of create the future of how to study medical devices, it's great that we had publications, but what was really important about this was that in the setting of a pandemic, and I would argue that in any setting where you put a product out and patients start using it and you only tested it in hundreds of relatively similar looking patients, we've got to get to the point where we have evidence hubs or active surveillance that we can have common language conversations with the CDC, with the FDA, with global regulatory bodies, and with other public health groups that need that information tomorrow, not in a year when we're kind of able to do a long study, write a paper, get it out in public. So, for example, in this case, a lot of these results that we were kind of churning out in the moment were able to inform things like the Tokyo Olympics and other public health events that were happening, where they needed to know what devices should we buy, how should we use the devices, how should we set up the diagnostic testing programs, and what other wearable devices can we use so that we can do effective contact tracing. So all of that information about how devices should be used and how effective they were was able to come out of these evidence hubs and kind of asking question after question in real time. The sports insights were far-reaching and impactful. As I said, we fed the results of them directly to the decision-making and regulatory bodies in the moment, and then eventually put them out into the literature. But that was a year later. So I always like to just take a minute to say, it's great to get the results out there so we can talk about them more publicly and have them under peer review. But it's really important as we set up medical device studies to get them under some momentum like active surveillance, so that we're actually able to take action and understand what we're seeing in the data in the moment and not when we get that out. Data quality is everything. That's a really boring 4-hour presentation that maybe we can do later, but we've got to make sure the quality is good. I just showed you the tip of the iceberg. Mackenzie's going to talk about the tip of the iceberg, but all of the time needs to be spent on the bottom part of that iceberg, making sure that the data is reliable, fit for purpose, high quality, complete, or we're not going to find out what we need to find out, and we're going to get the wrong answers. So all of these data were reviewed for quality to make sure that our analytics were reliable. And then this goes far beyond COVID. So I'm going to pass it over to Mackenzie now to kind of talk about that next data source. Wearables, and I think 20 different data sources pulled into that electronic health record hub. What you see here are some news headlines, because I don't know if she'll brag on herself, but a lot of the findings that we've pulled out of the injury and illness-based data set that she'll talk about are in the news. I think as we speak, we've seen a lot of reductions in terms of athlete injuries and a lot of success using this database, again, kind of in real time, in the moment, kind of within the month that we need to see those results. So I will pass it to her, and happy to take questions in between. That's never a good sign.
Not a good sign.
I'm gonna try slideshow.
Well, that's not good. Let's see if I can try to connect my computer.
I don't know. Or do we need to pass it? Do we need to pass it to someone else? Oh yeah. Or pause. Break time? This is a great time for a break. Okay.
Are there any questions? I can take questions too, Mary Beth, if we want to.
That's a great point.
All right. Thanks everyone for bearing with us with the unexpected break. I'm going to talk shortly about a couple of examples of how we use medical devices for sports research, like Christina talked about. I'll use 3 examples from our work that we do with the NFL and the NBA, so American football and basketball. And these are a little bit different. I'm going to start with an example that's sort of different than what we've been talking about, in the sense that we use devices to understand how to prevent injuries. So we weren't studying how the device was working, but actually using the device to be able to do the research that we were trying to do. And then I'll talk about 2 devices that we are trying to study the effectiveness in, with knee braces and with ankle tape. I just wanted to tee this up because I think all of us got into this space because we're interested in public health. And so musculoskeletal injury is really an important part of the public health picture and how we keep people physically active and how we keep them healthy. I'm not going to read through this, but just wanted to note that there are a ton of benefits of physical activity, and that these injuries can really impact your ability to be physically active. They can also lead to both long and short-term complications. And so this is a significant area for us to study, to really understand how we can keep people physically active and contribute to their health in the long term. From a sports perspective, and why I'm going to talk about lower extremity injuries today is because lower extremity injuries, or injuries to the lower body, have the highest burden to players and teams in sports. This is a paper from our NFL work. So in football, it's about 60% of our injuries are to the lower body. And then having this epidemiological knowledge really helps us understand how to target interventions for our clients that we're working with in the professional sports league. It also helps us understand where we should really focus efforts to help keep people on the playing field, or keep people out playing sports as those weekend warriors. And so the epi knowledges, and from the databases that Christina talked about, is really where we start. And then we've identified a couple of opportunities to really dig in deeper to either use medical devices to study these injuries, or to study how medical devices may prevent injuries or reduce the consequences of injuries. Christina talked about the linked databases that we use in sports. We're lucky that we've worked with the NFL and the NBA for a number of years now. I think we're in year 12 with NFL and year 10 with NBA. And so we have really high quality data that we can use to understand and reduce injuries from across both leagues. We're also a really unique area because we have a total population that is captured. So every player that comes into these sports, it's mandatory to collect data on all of them. And so we have the whole population at risk to study with these electronic health records. And then Christina showed this slide. This is really what we use as a foundation for our research. This is our injury and analytics database for the NFL. The NBA looks very similar, but today I'm going to talk about a couple of NFL studies, so we'll start with this. And we use this to really link together those data sources like Christina mentioned. The EHR gives us all of our health information about what the players experience, the injuries that they're sustaining, what their outcomes of those injuries are. But then we link in other data from game statistics or from wearable devices that I'll talk about today using GPS, so that we can understand the scenario or the environment that these players are participating in. So like I mentioned, I'm going to go through 3 examples. I'm going to start with the first, where we used wearables to understand how to reduce injuries to the lower extremity, particularly soft tissue strains like a hamstring strain or a quadriceps strain. Then I'll talk about how we're studying knee bracing and whether that could reduce injuries and whether those knee braces are effective in reducing injury. And then I'll talk about ankle taping and similarly whether ankle taping for practices in games could be useful for reducing injuries. So starting with wearables, Christina kind of teed this up. There are these unique devices that players wear that capture a ton of rich data about their participation. In games in the NFL, we have sensor systems that capture their games. And if you're a sports fan, maybe you see this on websites. You can see what players' passing was, what their distances were, what their high-speed efforts were. But in practices, we don't have that information. And so we had to find a way to understand what players were doing in practices and in their training, so that we could understand how to better prepare players for the game environment to reduce their injuries. So that was really the goal here, was find a way that we could capture data on players' experiences outside of games, so that we could understand how to train them better and ideally reduce their injury risk. What we needed the wearables for was to understand that non-game period of the season. And the reason why is if you're familiar with American football, our NFL season is a little bit unique. We have an off-season program, and then players go away for about a month. They have a month or so off, and then they come back and they have this training camp period where they start preparing for games. That training camp period is around 2 to 3 weeks before they get into preseason games, so it's pretty short. And that's the period that we have to really prepare them for the regular season and get them ready to go, and hopefully conditioned to the point that they're ready for the regular season game participation. What we know is that 2-week period, in that 2-week period, we see about 20% of our lower extremity strains occur across the whole season, happens in just that 2 to 3 weeks. So we identified from the epidemiological data that 1 in 4 players sustain a lower extremity strain. Those often keep them out for about a week. We found that 20% of those strains were happening in that 2 to 3 week period of training camp, and that there were spikes in the number of those injuries that happened right when they came back to participation. So right when they started participating again, and then right when they started contact introduction, so taking contact in the contact integration period. And so we knew we needed to do something to really understand why they were sustaining injuries during that period, and wearables seemed to be the right opportunity to get data on how are they training in those first 2 weeks of training camp so that we can guide how to better prepare for the season. And so what we did was we first asked teams to voluntarily provide their wearables data. We knew players were wearing these in practices, but they weren't required to. And we had teams that voluntarily sent all of their GPS data from their wearable devices to us to analyze data. From there, we performed a study to look at what can we see across players who get injured versus those who don't get injured that might tell us how we should better prepare for the season. And because of the initial findings, we were actually able to mandate that all players wear GPS devices going forward so that we can collect data on their training and preparation. And so interestingly, what we learned is when we take all that really granular data, we can look at accelerations, distance, speed. It turned out to be time that we needed, but the device was the only way to accurately get how long were they training and conditioning, how long were coaches having them on the field. So what we found was that time was something that we could guide and implement as an intervention to say, here's how long you should practice each day in those first week of training camp to reduce your injury risk. So from the device data, what we ended up implementing this year was, or last season, was a mandated ramp up, we called it, in terms of how you return to the season. We found from the devices that players and teams were returning from that off-season break in 5 different ways. There's 3 on the right of the screen here that were sort of, the red are sort of our worst case scenario where their first day back was their hardest day of practice, and then they kind of tapered off from there. We found that a lot of teams practice the same amount every day. Their players practice the same amount. But what we really found was that the clubs that use this gradual increase in the duration of their practice, or they use the high-low approach, which is in the yellow on the bottom right, those clubs had a 25% lower risk of injury occurring, not just in that first week, but across the whole season. And so we used that device data to then say, here, we can show you, if we compare all 32 clubs, here's the strategy that reduces injury. We implemented a mandated ramp up, so they have to increase their duration of practice across those first couple of days. And what we saw was a 25% reduction in lower extremity injuries in the first 2 to 3 weeks of training camp. So really an effective intervention that without the wearable devices, we wouldn't have been able to get at that period of the season and really figure out how to reduce injuries. There's more to come. There's a lot more information that we can gather from the wearable devices, and we're really excited to see how that will implement or how that will influence injury reduction strategies going forward. And that was sort of a first step and a way to really use this for a public health intervention. So switching gears, the 2 other examples that I want to go through are how we actually are evaluating medical devices to see how they could either improve return to play, reduce recurrence, improve care after an injury, or how they can reduce injuries. And I'll start with knee bracing. So there's a lot of different knee braces. There are rehab knee braces that people get right after they have surgery to protect their limb that had surgery on. So in this case, a knee. Usually they reduce motion, something like a straight leg brace so that your leg is held straight for a period of time. And so those are really used in the post-surgery phase. What we're looking at more here for our setting is how do we use prophylactic or functional braces to prevent injuries. And so prophylactic braces are those that are kind of off the shelf. You can go to the store and buy them at your local pharmacy. Functional braces are a little bit similar, but you can get them more tailored to the environment. They're more custom made. And so this is what we're evaluating now to understand how these braces could be used to reduce injury in our setting. Why do we need to do this? If you've watched football, you probably can imagine why we're interested in braces, because there's a lot of contact. It's a contact sport. But what's interesting is that right now in the sports environment, those players that are participating in college are wearing knee braces while they're participating. When they come to the NFL, to the elite level, we don't require it, and all of them take them off. And so the question is, does that actually help you reduce your risk of injury or not? And should we have them continue wearing these knee braces going forward when they're in the professional league? For the most part, we're focusing on linemen, and the reason why they're kind of at the front of the line, if you're not familiar with the football, they're taking a lot of the contact. And the reason why is because the way knee injuries, MCL tears in particular, occur to them for a lineman is they get this force to the outside of their knee, causes them to rupture the ligament on the inside of their knee. And the question is whether these knee braces can reduce that force or redistribute that force in a way that would prevent those injuries. Or prevent the severity of those injuries. And so Mary Beth talked about the process for how these types of devices are kind of brought to market, what we go through to evaluate the effectiveness. And that's not different here in terms of what we're looking to do in the NFL setting. We would start with something on more of the bench side with biomechanical engineering, understanding whether these braces are designed in a way that could reduce injuries. We would look at biomechanical efficacy in the lab, so seeing how they work inside of a lab environment. But then really where our role comes in and where epidemiology comes in is in that real world effectiveness. And this is what is the most important at this point for our setting, is understanding whether they actually work on the field and whether players are willing to wear them, what their experience is, whether they fit, whether they slip, what happens with them. And so right now in our setting, we are developing studies around these. We're putting together a plan to compare the braces that exist on the market and understand whether we think they could reduce the forces that we see in our game in football. But we're focusing more on those on-field injury rates and that real-world effectiveness to really get at, does the brace reduce injuries on the field? Does it reduce the severity or the time lost from the injury? And then what are players and patients willing to do? Are they willing to wear them? What issues do they have? Do we need to kind of go back and, as Mary Beth talked about, the iterations of the braces, do we need to work with the manufacturers to kind of iterate on that design so that it works better for our players and our patients? We don't know the answer yet. More to come on this. We are starting these studies now, and we're hoping that we understand in the next couple of years whether or not these can actually reduce knee injuries in football. And then the last example here is related to ankle taping. We're also looking at bracing and spatting. For those of you who might not be familiar with spatting, is where they actually tape over their shoe. So they tape their ankle but include the shoe inside of the tape. It's pretty unique to football. And so we're trying to understand, this is really something that has been popular among players. If you go into any athletic training room, or if you're interested in orthopedics, you'll always see players sitting on these benches with their athletic trainer taping their ankles. If you've played sports, you've probably taped their ankles. It's become so common that it's really just a part of their routine, but we don't actually know if it works. And so that is the goal here, is to understand how this actually works, whether you've had a prior injury and it's helping you not get a second injury, or whether it helps you actually prevent injury if you haven't had an injury to begin with. What's interesting about this is the reason that we're doing that is that we're hearing from high schools that they're spending tons of money on tape. And what we want them to do is spend money on things that, are actually effective and are going to reduce. We've put in a big push for things like AEDs on soccer fields, having an athletic trainer. So we feel like this is really an opportunity, even if we can't change the culture among our professional sports. If we can change it at the lower level, we might be able to kind of gear resources towards other settings. And so we have this opportunity at the elite level to understand whether or not this actually works to reduce injury. What we do, and this is where, again, epidemiology really came into play, because there's been a lot of studies on this, and they study only those players that get injured, whether they got taped or not. And so we don't actually know from the literature whether or not this works. So what we did as the epidemiology group working with the NFL is we implemented a data collection system for every player in every game. We collect before the game whether they got taped, whether they used an ankle brace, or whether they used spatting. And we specifically required that they collect this before the game, not if they get injured in the game, did they then get did they get taped to get them back in the game, but did they give this before the game? We found that about half of our players are using taping before a game or spatting. Bracing is very uncommon. Our suspicion is that bracing probably is more likely to reduce injury. Right now, it's looking like unlikely that taping is actually effective in terms of on-field performance. It stretches, does a lot of different things in the game. But we have another year of data collection coming up to really understand across the league whether or not these are effective. So we look at before the game, did they tape, brace, or spat? Did they then get injured in that game? And we compare whether their injury rates between whether they got these interventions or not. And so this is an exciting area for us. Like I said, it's exciting because it's translational to other areas of sport. It's also a question that so many people have asked and that we haven't seen great study designs to really address. And so this will be a fun one for us to figure out in the next year or so, whether these can help us and then really guide from there, maybe identifying other opportunities to reduce ankle injuries if taping doesn't work. Is there something else we could do to kind of reduce those injuries? And I'll close there, just a couple of examples. I think if you're interested in orthopedics, as Mary Beth said, there's a ton of opportunity. There's so many things that we need to understand whether they're effective or not, and there are not a lot of epidemiologists working in this space. And so what we get are taping studies that evaluate only the injured players and whether they taped or not. And so please reach out if you're interested in this area. There's so much work to do here, and we would love more friends involved. Any questions or thoughts or comments on that? Yeah. Yeah. Mm-hmm. Yeah, it's a great question. So when we launched so our wearable devices in sports are a little bit different than I think what we think about in normal life. We think about like Apple Watches, or Christina mentioned the rings. Ours are actually worn by the players. And what we did at the beginning for both leagues actually is we went through an evaluation process of how do they work, how can they be kind of embedded into the equipment in a way that will give us reliable data, how consistent are they across devices. So with NFL, at least, we have multiple devices that are used. How can we kind of aggregate them? And so from the player's perspective, they were really part of the initial evaluation to see how can they be on your body in a way that doesn't interfere. For us, they're embedded in their jerseys in most cases, or they're worn. And so we get that piece of it. And then from there, we evaluated on the data side with our engineering team whether or not they were sort of equally effective across devices. And it was definitely a long-term process. It took several years before we got to this point where we can really meaningfully analyze the data and feel comfortable that it is representative of what they're truly experiencing. We still have some crazy things where we have a player turn on their device 4 hours before their practice and then they walk around, or the devices die, the battery dies 4 hours before or something like that. So there's definitely some user interface things that we need to work on, but it's a continuous evaluation of those factors. So yeah, great. It's a great point. Yeah. Yeah. Mm-hmm. Mm-hmm. Yeah. Yes. Mm-hmm. Yeah. Yes, it does cover everything. It's a great point, though, that right now in the NFL, if you wear a brace, it's probably for a reason. So we definitely have, confounding my indication, they've mostly are wearing it because they've had a prior surgery or they've had a prior injury. We can adjust for that based on the data that we have available. Luckily for us in the NCAA, they're required to wear it no matter what. And so we can kind of use other levels of football to understand if it's mandated and you're required to wear it for any reason, how does that work compared to in our environment where you aren't required? But yeah, it's essentially collecting the data. And like I said, we're extremely lucky that we work in a closed population and that we have mandated data collection. I recognize that's not the case anywhere else. It's partly why, we're able to translate information to other areas, because we have that ability to collect really rich information about why are they wearing it, what are they doing, what else is happening in their environment. Yeah. Yes. Yeah. Mm-hmm. Yeah. Yeah. Yeah, we have some interesting literature that we've put out where we can actually show, if you don't invest in that data set, that how you can get very biased results very quickly. So we're very fortunate, and we know we're fortunate in that sense. And like I said, the hope is that we can use it for other settings as well and really help others understand the efficacy of these and effectiveness of these devices.
Let's pause here. Are there questions? We will have the panel coming up. Jot them down, and we'll let David talk first, and then you'll have all the questions for him too.
Great.
Okay, so thanks everybody for coming here. This on, or am I close enough? All right. So thanks everybody for coming here. Some disclosures to start. No specific funding was received for developing this presentation. In the past 12 months, we have been performing contracted services for medical device and pharmaceutical companies, but we have no stock ownership for the work that is presented here. Here is some contact information with some QR codes, because we like to collect data all the time. So I guess we'll see how that works out. I'll give people a second just in case they would like to take a snapshot of this. This is what the course objectives are, to kind of just remind myself what we're here to kind of talk about. Obviously, I'm not covering all of these things. We're exploring the methodological and data source considerations for research involving medical device and combination products. As you've been hearing across this, we have a lot of real-world examples, and some of them are very, very interesting and related to some of the stuff that you'll hear that I'm going to present. There is definitely a dynamic regulatory environment that is changing all the time, so I'm very glad that we have Mary Beth here to help keep us on track with that, but it is very, very fast-paced, and some of my talk will also overlap with that as well. You heard a little bit about the science of patient input. That's very, very important, and you're going to hear more about some real-world medical devices. My talk, as Mary Beth alluded to earlier, I'm an applied statistician, but I am very excited to give this talk because a lot of the things that we do is a little bit different than what other statisticians are doing, and it's much more in line with what you are all doing in the epi space. We borrowed a lot of the things from that field and brought it into clinical trials. So there's not many statisticians who actually think as much in that world. There needs to be a lot more because of how fast-paced and all the types of changes that are occurring. This slide is, or these sets of slides I'm going to give are going to talk through and give you the background of how I think as an applied statistician, why you're very, very capable of entering in this space. We want you to enter in this space so you can push forward in this space, and we absolutely need that. There's not enough statisticians who can think the way that you all can in this room. So I'm hopeful that at the end of this, you'll join us on this side. There's a couple other presenters here. You obviously see this. I had to swap out the first logo here from the FDA to Mary Beth's new employer. Congratulations, Mary Beth. We have some very, very large companies here. Our company is on the opposite side of the spectrum from IQVIA and J&J. And I'm going to talk a little bit about that and what that means Really, to get down to it, as was talked about earlier, medical device companies are very, very small. It may be 2 or 3 women and men in a room who are starting up, and it's the CEO, because one of them has to be called that because they just started an LLC, and here we go. It is very, very different than the pharma side. There's a lot of fast iteration, which I'll talk about. So my lens and where I've gained all of my experience is closely working with those very small companies. Being another small company, a lot of the tactics that we take in trying to push study designs forward come from the way we interact with all our clients. Is they're iterating fast, we're iterating fast, we have to do that together. We have a lot of allowances and desires to keep budgets down, and a lot of constraints that they're having to deal with necessarily go into study design. So we've really learned a lot, and it's very similar to some of the bigger companies, but it's a little bit different because of how closely aligned we are with these small companies who are developing these products. Here's an agenda. I had a similar type of agenda slide last year. It's morphed a little bit, so this is pretty relevant, and it at least gives me some guide markers to know where I am in this deck. So I'm going to get into some things you were taught, practice, and advocate for. What I'm about to talk about is RCTs are still relevant. There are places when they absolutely cannot be used for reasons like in the NFL or other places where there's not a way to do that. There's other ethical reasons that you cannot do this. Mary Beth noted in one of her slides earlier that when you have surgical trials, it may not be ethical to actually have a sham surgery procedure. A very interesting component with that is a couple of years ago, some of the implantations our world is in spine. Over the past decade, we have done probably over 85% of all the spine PMAs. So these are very, very high risk. They're all class 3 devices. They all have clinical data. They all have long-term post-approval studies. But that's really where our sweet spot is. In that space, though, what is currently developing is less invasive procedures. When you go from, for example, if you're going to have a fusion in your spine to a less invasive procedure, the control group has to change. So again, nobody's going to do a study design and go to FDA and say, hey, let's do this study. Let's put somebody under anesthesia and then actually not do anything to them. That's not going to occur. However, when you see people going from mechanically implanted devices and real metal things that are going to live in your body forever to something that is a lot less invasive and can be done either through a needle or otherwise, the control group has to change. And what you want to get is somebody who is really doing conservative care. So is there a blinding aspect of it? Yeah, we are attempting to do that. You definitely can do sham radiographic evaluations so that the patients don't know. You can separate the providers and have different doctors doing it. So some of those key elements that you can get out of the more standard pharma trials, we are looking at very, very heavily. And part of it is a big push of less invasive procedures. So for a long time, there was very, very seriously invasive procedures. Again, there's a shift because of technology improving. So we are getting back to trying to replicate some of the pure randomized trials that you would expect in other spaces. I have another disclosure. I don't play video games, but I did need a set of compelling slides. So you'll see in the next one, and I'll reference back to this, what I mean by that. So RCTs are the bedrock of most regulatory submissions, and they will continue to be. I do not expect that to totally go away. I saw this and I said, hey, that is a very, very good thing to post on here. Last year, I may not have done this, but even in the last year, there's been so much progress. And so I felt comfortable putting the Flintstones up there with all the things that may allude back to. Again, RCTs are not going away. But there is a very, very strong advocacy from FDA, from industry, and collaboratively with the FDA and industry together to design more innovative trials to answer questions that would not be possible if it were not for the ability to use real-world evidence across the board. This is just reiterating what I just said. So the main takeaway here is going to be that, again, RCTs can't answer all questions, or they have very, very large limitations, so large that it may make them not able to answer the question that actually matters. Some things that you'll get into is some of the surveillance and long-term studies. What is different and was talked about earlier is that when you implant something, the expectation is going to be that it's going to be in the person for the life of that particular patient. That's going to be a really, really long time. So there's a totally different thing in the device space than the pharma space for lots of different reasons. This is going to be some background that you all will kind of know. Randomization has a lot of great features. It simplifies a lot of the analyses that we have to do, obviously. Then exposure is the only difference in a purely randomized trial. There's always nuances of how you do randomization and permuted blocks, all those different things that you could do when you learn in Stat 102. Yeah, that's still there. But because there's so many limitations, we've really pushed ahead. On the bottom of this slide, it talks about they're prohibitively expensive. At the end of this talk, I'm going to talk a little bit about some of the things that we're working on that alleviate that, but that is a real constraint. We're dealing with companies who they go out and they have to do fundraising rounds, and so they'll go over to the VCs and they will have real constraints that either may make it so that they cannot get that trial funded, and on the other side, they may have 50 million that they just raised to be able to do this trial, but there's still an advocacy for, hey, this better not take a really long time, and part of that is because of how fast these devices change. There's engineers and their R&D departments are always developing. If you start a trial and then fundamentally change the device in the middle of it, you very well may have to start again, and that can be a killer for these size companies. So again, I'm going to reference the video games a couple of times for no other reason than I think it's compelling. We had trials that were very, very simplistic, and that's represented by like a Gen 1 on the left side of the screen. The main takeaway of this slide is that we're already at the next, and even from last year to this year, the amount of progress that we see and are able to implement into studies is huge, where every day there's new allowances. Part of that is that FDA turnover is non-trivial, but there's a very good thing is that they're bringing in new staff who have different philosophies and are more willing to collaborate. Some of the best collaborators at FDA are people who've been there for a really long time and are really well versed in the new and necessary study design considerations in the medical device side. We have a lot more tolerance and flexibility on the medical device side than in the pharma side, and that's a really, really good thing. It's absolutely necessary that we have that because, again, you would not be able to get trials done if it were not for the ability to be innovative. I already talked about a little bit of these, but here's just some of the things. There again, it's very, very fast iteration cycles, and it may just be the engineering side. The other very real component is the amount of competitors that these medical device companies have. It is not that you have one particular molecule and you have patents on it for a really, really long time. There's a lot of copycat designs, and you have to be very, very fast to market. That is essential for all of these companies. The availability of the data, which I'm going to talk a lot about later, some of the generational designs and some of the features that come out of other trials previously, they may be applicable. And so we have the space to pull in historical data in ways that are not accessible in other areas. As talked about earlier, and what is really important, you're going to see in the next set of slides while I walk through and kind of do a little live data interpretation, is there's real operator effects and there's real failure modes that may result in having revision surgeries and may have other mechanisms that will pull things out. The endpoints are really important in medical device trials. They're broad. They're going to require, in many cases, radiographic data. That can be a real constraint sometimes, but in the US especially, you cannot get an orthopedic product through generally without having radiographic evaluations. And it's not just one radiographic evaluation. It's going to be baseline. It's going to be all the follow-ups. It'll be CT, MRI, X-ray, all the modalities will be needed. So there's an extensive portion of our trials that involve radiographic data. This I showed last year, and I'm going to walk through this. Generally, this is kind of the type of data set that we're going to see. So I can actually visualize how many patients are in this trial because it's not thousands, it's not tens of thousands, it's going to be several hundred, and that's the size of the data set. You're going to have one primary patient-reported outcome, and you're going to have a lot of different sparseness. You're going to have a lot of different operator effects. This is sorted by site, and so there's actually like 10 different sites in here, and as you scan across, you can actually see that patient trajectories are different. That is going to be for a couple different reasons. One of them is that the sites that are in here, when I really study this, you can see some may be in a surgery center and some may be in a hospital or academic setting. You can actually see that in here. You can see that as how close or how much variability there is, how there's some surgery centers that are getting people out the door in less than 24 hours. There's some that are getting people out the door in a lot more time. So when you actually look at this data set, it's entirely digestible. It's a very, very clean outcome. There are trajectories in here that are truncated. They're truncated because the implant is now taken out. So that's not necessarily missing data. We've generally solved that, because when you go to FDA, they're going to require any patient who has a revision to be considered a failure. So we've simplified a lot of the ways we have to analyze these trials. But again, the big takeaway of this is these are small data sets. And at the end of this talk, I'm going to talk about what that size of the data set allows us to do when we need to be a little bit more creative and bring in different kinds of control methodology. I'm going to get into some methods and technology. I'm going to talk through things that I think a lot of people in this room are going to be well versed at. It's going to be propensity score. It'll be some Bayesian hierarchical modeling, which may be a little bit less known, but generally understood. And just to have you all exposed to the way that I'm thinking, because what I think that you will take away from this is that a lot of the things that you're doing and thinking about are very much implemented on the medical device side, a lot less so in the pharma side, but absolutely there are sets of statisticians working with these type of trials. We have a lot of flexibility, as I talked about earlier, and so one size does not fit all. There's a lot of ways to solve these problems. Specifically, what I mean is you may have a company who comes in, says, hey, we have this amount of money to do this type of product. This is our only product. How can we get this done? And we will have to collaborate with FDA on what is the right trial design. What is very, very nice working in the medical device side is that FDA is very open to collaborate with you. They will help with study design considerations. They understand and have been through this process a lot. We have a lot of exposure personally at our company, but just across the board, you'll see that the teams that are working on the FDA side have seen innovative trial designs, are willing to push the boundaries. There's guidance documents that they published. Greg Campbell recently passed away, but absolutely pushed forward greatly all of the different Bayesian techniques that are currently in use. And you can see currently that some of them are trickling into the pharma side just now. These have been here, and it was 2010 guidance document or so that actually came out and really pushed forward how Bayesian statistics can be used in the medical device side. I will be transparent that there's a little bit of a backslide on the medical device side in terms of some of the Bayesian techniques that are being allowed. And it's specifically that comes in adaptive designs and the ways we can use predictive probabilities. There's lots of different reasons for that I can discuss in a different talk. The obvious question in these trials is compared to what? There's a lot of ways to cut these, as I was just getting at. Do you have patient level data is going to be one consideration. Do you have a patient level control, population control? I'll talk about both of those later. What if you have a hybrid? What if you have literature, very, very good, robust literature, and you have patient level data? How can we combine those sources to get out a single control group to allow labeling just as if it was a randomized trial? What if you don't have any of these and you still need a comparator? To get on the market in the US, you have to obviously compare to something meaningful. I will leave specifically defining meaningful to Mary Beth, but generally you really absolutely have to compare to something, but there's just a lot of things that you may be able to compare to. I broke this talk, this is going to go into patient level and non-patient level data. I'm going to talk about both. The combination I can briefly talk about at the end. The ways I'm going to go through this is I'm going to focus on propensity score matching, again, just to give you exposure to the way that I'm thinking about it, because I know that a lot of people in this room know this material as well. But for those that do not, I will kind of go through this set of slides just to make sure that you're in tune with the ways that we're thinking about it and in an applied setting where we're focusing in on what you need to know. In our world, sometimes it's impossible or not feasible to randomize. One application of that you would see is that when you have a surgical procedure that's been on the market for a long time, it's very well known, you may actually have surgeons say, I'm not going to do that anymore because, hey, I've been practicing in my practice. I've been using a device for 10 years. I'm not going to go backwards. I do not want to destroy, for example, I don't want to destroy motion and fuse bone. And so they absolutely, and as a practice policy, will only implant devices rather than fuse bone. When that happens and FDA has an expectation of, hey, go up and have a control group of fusion, and you can't find enough surgeons to actually do that procedure, what do? You have to do something about that. And that's sort of the one of the originations of where some of these pressing models came from. It's that you could maybe get patients to go into a study and get randomized. You can't get the operators to do it because there's accepted technologies already on the market. When that occurs, you are forced to either not innovate in that space or do something more creative. And that's really many, many years ago where this pushed forward. I'll give a little bit of background on what that means is that we have multiple PMA studies that have gone through that have borrowed historical control, patient-level data, use propensity score matching, have gone on to have regulatory approvals. So FDA has seen those or exposed to them. We have many trials ongoing right now using different sets of technology. There's references at the end you can look at. The founder of our company actually created one of the heuristics that we use routinely for propensity score matching. But again, we're solving problems where it's not that we're only trying to be efficient, it's that we have to get these trials done or not innovate in that space. So it's a very, very real problem that we have right now. Propensity score matching essentially is we want all the features of a randomized trial. The simplicity of those analyses are obvious to everybody here. And we want to be able to get inference, and specifically we want to be able to get an FDA label that looks and smells and tastes and feels like it just came out of a randomized trial. We're able to do that to the satisfaction of FDA multiple times, and we're currently doing that multiple times. Mary Beth said that there may be an equation, so here's your one equation that is on the slide in this gray box. And again, as I was speaking through, basically what you want to get is subjects that have identical values of being treated. There's variations to that, obviously, that we can go into in a different talk. The key assumption in all this is that you have no unobserved confounders. I'm going to talk a little bit later about some of the variables that are absolutely necessary to have. You can't use this technology everywhere. I have a slide at the very end that's going to actually walk you through the process of what an FDA submission looks like. When I gave this slide that I'm about to get to later, people were surprised at how much collaboration there is with FDA. Again, as you look through these, remember that this is, if you're doing study design considerations, either having us help you, other people in this room help you, doing it yourself, FDA is willing, open, and you absolutely want to engage them very, very early. I'm going to advocate that a little bit later in the talk. Here's a visual of essentially what was on that last slide. You can do propensity score matching in lots of different ways. We prefer subclassification. FDA strongly prefers subclassification versus other methods. That may be a little bit less known to the people in this room who may be more inclined to think about inverse weighting and those type of models. FDA does not have a lot of experience in evaluating inverse weighting in applications that go on for approval. They absolutely do in subclassification. Subclassification is depicted on the bottom right of this screen, where you're essentially getting 5 randomized trials. You're splitting that whole data set, and then you can evaluate them similarly as if each one was a randomized trial and then collapse them together after testing for it. What is interesting in the left side of this screen is that there are a couple red dots, and I'll talk a little bit about what those mean in our world, and I'll rope it back in at the end. When you go, for example, and you purchase a historical control data set, or you have a historical control data set, the goal is to say, hey, I have this new product. It's going to have inclusion, exclusion criteria, X, Y, and Z. Your control data set may have a very, very different set of subjects in there. If they're outside the inclusion, exclusion criteria in the control arm, they can be trimmed off the bat and discarded from the data set without any kind of error or without any kind of penalty. They're simply not applicable to your inferential scope that you're going for. FDA is fine with that. FDA is absolutely not fine with trimming on the treated side. And so you can see there's purposely no red dots in that side. It is very difficult to justify at FDA to ever exclude anybody who's ever been had any interaction with the device. It will be a discussion that I would rather never have with FDA. On the control side, it's very different. If they don't meet the criteria, you can simply drop them from the data set. At the very end, I'm going to rope back in about some complexities in that, in what happens when you have control data sets that have 40% of red dots. What does that mean in terms of people acquiring that data? What does it mean about the data in particular? But that's the big thing to get away from this. I've already talked, there's subclass, matching, weighting, adjustments. There's lots of different ways we can do propensity score matching. There's lots of talks going on at this conference, and there's people in this room who can really help fill that in. But from an applied standpoint, you can see the things that we need to get out of this and why you are very, very capable of entering this space and helping out with this. I talked about variable considerations, but here's some specifics on this. The bottom part of the slide is saying that you need to make sure you have all the variables that matter. FDA is not just going to say, hey, can we get the mean ages the same and we're good to go? No, that's not going to work. It needs to be a robust set of data across many, many variables, and some of them may be radiographic parameters, which I'll talk about later. But you need a lot of data to be able to do this well and have it be accepted by FDA. When you have that amount of data, though, you really can get this process done. We've been successful at it many, many times. And FDA is continuing to evolve in their acceptance and knowledge and institutional knowledge of how these designs work. Obviously, we're only able to balance on what is observed. There's obviously the key assumption that the unobserved are not very different. Let's go on to confounding and bias, which these things are going to be repetitive from what you heard earlier because these are the core things that we have to talk about. Confounding by indication, healthy user bias, confounding by frailty or comorbidity, healthy adherer bias, and then there's some additional text on the screen. This room is very, very already well aware of what you need to be on the lookout for when you have two different data sources, why potential bias can creep in, and it may creep into a point where the data set is not usable. A lot of times it is overcomeable and able to be used, and part of that is because of the source of the data. In medical device trials, especially in our space in orthopedics, they've been standardized to a point that you can go from data set to data set, and generally you're looking at the same type of patients. Even without any kind of adjustment, you're going to have a lot of similarity across them. That's not the same in other spaces, but part of the reason is that the inclusion-exclusion criteria in the studies that we deal with have been so standardized, and FDA has reviewed them so many times, that we have a general expectation of we're able to deal with these type of things. We're really already kind of in the ballpark, and that we've capitalized on that greatly. And it's present not just in spine, but across the board. It's not just in orthopedics, it's broader than that. It's trickling into diagnostics, which I'm going to talk about just briefly and kind of get into. But when you have more and more high quality data sets that have been FDA reviewed, the opportunities to do these type of designs continues to grow, and I expect that they will continue in that way as FDA has more exposure. Here's some general study design considerations. The key feature is that you're not using outcome data anywhere. We have to go through a lot of rigorous audit-proof documentation that shows how we bifurcated the data set so that the statistician performing the analysis has no access to outcomes. So it's not just you read that in the book and it's kind of important. No, it needs to be there. You have to be prepared that you may get audited, and you need to show that documentation. And so it is a very, very central portion of any kind of business when you're doing this. You need to make sure that IT is involved. Like, there are very, very real mechanisms that if you want to discuss, I'm happy to discuss afterwards about how to work through that documentation about the firewalls. When you break the outcomes, that obviously requires a larger team. You're going to need one team blinded, one team unblinded. You can do that in one company, two companies, three companies. There's lots of ways to split that out. The key thing to take away is don't overlook this and just say, hey, I just kind of split it in half and used just the baseline data in my model, and it was just sitting there. I just happened to sort it and just didn't take those rows. It's not anything like that. It's a very, very real bifurcation of the data and then documentation of a difference in those different data sets. You may or may not have seen this. This is a love plot, and this is the diagnostics before and after matching. And you can see that there's dark blue dots that are showing very, very large differences between the groups after an implementation of a heuristic. This happened to be the heuristic that our founder created with Don Rubin, who is the co-inventor of propensity score matching. You can see that the light blue dots are right in the middle. We were able to take this kind of demonstration. This is one of the key tools to demonstrate to FDA that you've sufficiently dealt with the matching aspects of this. But when you do study designs like this, you're going to go to FDA, and you'll see later, you have to lay out the criteria that you're going to use to eventually evaluate this. So it's not that, hey, you do a bunch of different models, and then whatever works, you can go to FDA and say, hey, look what I got. FDA has seen the criteria that you'll eventually use before you ever do it. They're going to look at it along the way. Again, it's a very highly collaborative process, which I had some feedback last year that was surprising to some of the people in the pharma space. And it's absolutely, and I can keep looking at Mary Beth because I know that she will definitely stand up and say, yep, FDA is on board, be collaborative, be open and transparent about it. I'm going to transition a little bit now to say how different is real world data from what you can get out of an RCT. We've seen the spectrum. Sometimes it's not very, very different at all. Sometimes it's fundamentally different. And so it's not very clear cut when that's going to occur. So even though I was talking earlier that you can take some standardized trials that have inclusion and exclusion criteria and lots of different features that should make the data set similar, you can have wildly different data sets depending on what you're looking at. So you really have to do a lot of deep dives. What I'm going to do at the end is I'm going to talk through how we work through them, and then I'm going to advocate for you all helping us on the statistical side work through them. You have a lot of the tools that we need to be able to expose where biases are, when they're overcomable, when they're not overcomable. That's what you all think about all the time. I'm going to briefly talk about how we are starting to repurpose some of those propensity score matching technologies to kind of characterize, quantify some differences in the groups. So what I talked about earlier in propensity score matching, if there's an intermediate step that you can go through to kind of start describing to FDA, to the clients, to everybody, how different data sources are. We have a couple ongoing projects where we aren't using propensity score matching for the final analysis. What we are using propensity score matching for is evaluating how different the data sets are. And so if you were able to run through a process and create a love plot like the one I just showed you, and the initial is not very different, or it's really, really different, that may mean a lot. It may mean a lot in what your regulatory pathway is going to be. It may be that you can use this data source or not off the bat. It may be used as a sensitivity analysis, but some of the tools that you've already been learning, we've repurposed and are using them in new ways now that FDA and industry has more and more maturity in these type of algorithms and the ability to eventually lead to regulatory approval. I'm going to go through when you don't have patient level data, and that's a whole other side, but it's really, really important. What I have up here is a diagnostic tool that was looking at bone density. And so what people will normally do is you'll go to the doctor before you have surgery and they have to measure this. They may measure this on the basis of different risk factors. This tool is in place of a more invasive procedure to get out how hard your bones are. There's no way to randomize, there's nothing to randomize to, so we needed to do something totally different. The red line would represent a collaborated and negotiated study success criteria. And then the left side of this graph is the posterior probability based on the data collected within that study. Performance goals are one way when you don't have patient level data that you can get approval. Mary Beth can expand on, and other people in this room can greatly expand on the differences and the implications on labeling. There are very real differences when you take a fixed target and say, let's compare against it. But when you don't have patient level data, and sometimes the best that you can do is if you can come to an acceptance criteria, which may mirror a little bit more what they're doing in EU to an extent. Here's one way that you can get study designs done. The other side of that coin is when you don't have patient level data, but you don't want to go on a performance goal route, but maybe you have many different data sources, for example, from literature. You have to be able to combine that data in a meaningful way and specifically articulate and characterize how much heterogeneity is within that set, and then how you actually collapse that back and then talk about how much the differences between the data sources should impact your eventual success. And I have a couple sets of slides that'll go through that. I do want to talk about, again, just to have you exposed to the way that we're thinking about this. Exchangeability is obviously a very important component of this. If you have different data sources, you're making an assumption that the data are exchangeable. That means that we could swap all the labels and it's not going to matter. There's FDA guidance in this set of bullet points we use, but it's really grown out of different reviews of FDA guidance. And these are the things that you're going to look through, and you're going to need to be able to articulate that you have considered them at the very least, and then manage them will make it much more likely you can go on for approval. You can't do anything without quality data, so that's obviously bullet number one. You can review inclusion-exclusion criteria. You'll look at the baseline demographics, the preoperative status, and you need the same clinical endpoints. It's not necessarily the case that you need identical endpoints, and there may be different PROs that are highly similar and can be shown to have such a large correlation that it is good enough for what you're trying to accomplish. But you obviously need to have data sets that are rich enough. In our world, as I talked about earlier, it's not just a single outcome. You're going to have something like a patient-reported outcome, radiographic parameter, a safety parameter. So the actual endpoints that we're looking at are multi-pronged. If you have data sources that don't have all those prongs or only have partial prongs, you have to do something different. So when you're going through and reviewing this type of data, this is the type of things you should think about even before you do any kind of analysis. Time is a really important component, one that is focused on all the time. This is a real study. On the top is the investigational arm. On the bottom is the control group. Not all data sources look like this. You can see that it's indexed by 2016 to 2018 along the bottom. You may have differences in time. This one's great because you can see, and I'm not hiding anything, it's just kind of looked a little prettier, so I put it on the slide like this. There's a lot of overlap here. sometimes you have data sources that are not overlapped like this at all, and they really have differences in time. The difference in time is going to get back to the operator effect. There are real changes in the practice of medicine, and the changes in operator effects can greatly vary even by a couple of years. Sometimes there may be a conference, there's new techniques that they teach, and now fundamentally you're going to have outcomes that are much, much better. If you have 70, 80% success rate, now it goes to 95%, that may mean something very, very different. When we're talking about aligning studies, it's important to recognize how much a time impact will matter. A couple slides here that will just illustrate what I mean by Bayesian hierarchical modeling in case people aren't aware of this. This is an illustrative example. In this study, there were 20 or so literature articles. This was a formal PRISMA literature review was complete. So you go through all the rigorous criteria of that. We don't perform them. Our collaborators did that, but they came back and they said, here are 20 publications of all of this data. There's a response rate on the bottom, and it goes from 0 to 1. You can see that it's about a 92% success rate when we're all done. The dark blue points are after shrinkage, and so you can think of a random effects model. At the end of the day, what you're trying to get is the right estimated rate out of these 20 articles, and then you're going to end up using that in this way. So if you're successful in combining all of that data, you're going to end up with something like a posterior probability. We can talk about lots of different ways to formulate it, but you're going to have a set of data that is indexed by how much information is in the literature, and that will dictate how high this graph goes or how steep or narrow it goes. But again, it was about a 92% success rate. Having this versus what I showed earlier with a fixed line, that red line is a single performance goal fixed target. That's one side. The other side is this, where you actually have variability. When you have variability, what you can do with that is a lot different than a performance goal. It means it has very large implications what you can get onto a label eventually. This is a slide that is built off the last two, but it's just how we interact and are talking when we're talking with clients about designing these studies. There's the treatment and control, and then the group difference on the bottom row. This is simulated data for the treatment effect because this trial didn't occur. But the process was, and they went out, they did the evaluation, they understood, hey, look, there's a control rate. We're able to get this data aggregated like this. Here's an expectation of our rates. What's the group difference? This was a non-inferiority type study. So this is entirely acceptable. You'd be looking for how much that posterior distribution is above or below minus 0.1. This would absolutely satisfy that criterion. You'd be good to go. Again, it's simulated data, but just to give you some exposure to the type of ways that we are interacting. And so when you think about literature controls, you have to go through this. It's not, hey, and we see these, but ideally you go out, you have your control group, then you do your trial, and then you compare against it. And it's really done in that sequence. The other side is, hey, we have all our data, now let's go back and do the control group and then do that. There's approvals based on that, but in an ideal world, you would do it more like this. And I feel like if I'm up here talking with you, I should show you the better way to do that because you may not know. I'm going to go through some things about what I'm calling the process. Whenever we're presented with any kind of problem, these are the sets of questions that we're going to do. They all boil down to make sure that you're critically planning this trial, which is what I was just talking about in the last set of slides. Again, there may be reasons why you have more or less data available to use as a control arm. There may be more or less data for the investigational arm. And by that, I mean, sometimes it can be prohibitively expensive, or it may not be necessary to have entire investigational arm data sets. So instead of going out and getting 200 patients on the investigational arm, you may be able to go out and get 50 and then take 150 from or more from your previous trials and then combine data sources. So your patient level and then something like a literature based on the investigational arm in the same way that I was showing you how you could combine on the control side. You may need to do that because you have a slight generational increment. And again, this may be a very small tweak that can be demonstrated on the bench to a degree, but there may be some remaining uncertainty that you need to overcome by having a small supplemented data set of even the investigational arm. So really understanding what constraints you have is really, really important. Again, this is in contrast on the pharma side where they have different abilities to enroll different size trials and different levels of resources. These are very, very small medical device companies, and they have levels of constraints that are very real that can maybe shut them down and they're not able to go on with that product line, which doesn't just mean product line, it means that's the company, that's it, that they can't go forward. So we're solving do or die problems for these companies. I've been talking about this all day. How do you meaningfully get groups for comparison to ultimately support regulatory approval? That is the name of the game. That's what we do. That's why at the beginning of this, I was introduced as, hey, look, he's an applied statistician, so hopefully this keeps you grounded. This is what we're focused on all the time. It is ultimately to get to this. There's some very, very interesting things that we heard earlier that are coming out of the NFL, things that we're definitely going to go borrow, and we do that all the time, is hear some of those things and then try to rope them back. But this is our goal, this is our mission, this is what we have to do. So, but we're going to pull ideas from everywhere. I talked earlier about giving you a little bit of background about what this looks like as part of the process and how collaborative FDA is willing to be, but it is essential to be very early, transparent, and consistent in your communication with FDA. You do not want to be on the side that is trying to just do something at the last minute and throw it in there. Absolutely, don't try that. You will have a very rough time with FDA. However, this is the general structure of this. Later on, I'm going to insert another box in here that's going to expand on this. But generally, as I'm talking about, you'll be presented with a problem. You'll learn about all the different kinds of constraints. You're going to end up with a critical assessment of the data. You would immediately have an FDA pre-submission. An FDA pre-submission is not like you're submitting anything like on the pharma side. These are very, very short sprint pre-subs. I recommend that somebody ask Mary Beth about what a sprint pre-sub is, so she can articulate a little bit about that, and I can help talk about it from my lens. But these are very, very short, very fast meetings with FDA as you're still trying to iron out study design considerations. It's not like you go through the whole process and you get to a fully baked idea. FDA wants you to engage early and often and transparently. You want to say, hey, we are contemplating this type of study design. What do you think about it? Does this seem like a pathway forward? The reason you would do that is because, again, these are companies who, if you go too far along and you fully bake a study design without getting FDA buy-in, they may have expended so many resources and have so many things in motion already for FDA to come back and say, that's not the way to go. That can be very detrimental to them. So you really want to interact early, transparent. Often do not be scared of it. There are people in this room who can absolutely help you through that if you've never done that before. The other interesting and very important part of this is you can see at the very end of this is saying you unblind the PS statistician. That's what I meant by you really have to demonstrate and document the difference in outcome data and baseline data. You really have to separate them and keep that data separated through the entire life cycle. It is not uncommon that you may get a statistical model that works, that can align the groups after propensity score matching or otherwise. And FDA, during the review process, despite having seen it before, may ask for a tweak on that model. You can't do that if you've already exposed one of the statisticians to that data. So it's absolutely critical that you separate that, and for the whole life cycle of that product, keep everything separated until there is a decision either way, approval or not, to maintain the ability to agilely respond to FDA's questions, because you should expect that you'll get some. The rest of this says that there's an interaction with FDA here, there's another interaction here, there's another interaction here. I'm just trying to distill into you that you will, in any type of these more innovative designs, have a lot of collaboration with FDA. I have this in the next slide that are just some basic milestones that is kind of a bulletized list of what was before. You can read what's on the right side of this chart, but you're going to see on the left side that before you start the study, you're going to have interactions. Following an enrollment and a data lock, you're going to have another set of interactions. I'll pause here for just another moment. You want to do this as soon as enrollment is complete. You want to do that before there's any outcomes. And things that we routinely do, you're going to have 6, 12, 24, or longer month primary endpoints. That means that if you can interact with FDA and get agreement on a study design, you can do that before there are any outcomes on the investigational arm. That is a really important step to take when you're trying to replicate what happens in a randomized trial. Here's the following ones, which again are just going to say they're going to have a lot of interaction. You have all these slides, so you can kind of read through them. I want to talk a little bit about the different data sources, and then I'm going to go into two particular parts, which is on the safety and the radiographic side. We do have Obviously, all this is going to come in some kind of EDC. There's also going to be medical health records that we can borrow on. We have a lot of things that are ongoing with claims data, and we are able to use those in some ways. Some of the bone growth things we're able to use, and so these will be relatively large, not as large as Mackenzie said, 600 or 700,000 million billion data points, but there's a lot of data in there. That's pretty good for what we're doing. It may be tens of thousands that we can pull from and do the same processes earlier and construct data sets that are meaningful for various things that we're evaluating. Uh-oh.
So it sounds like there might be just electronic or something. Yeah. Is it only electronically monitored? Things like
I'll do a lap and I'll let you know if I see any smoke or fire. Now that everybody's awake again, had a moment to stretch. 5 minutes.
Paul, I thought you said you were going to like nudge me if I needed to speed up a little bit. Not that.
It's so exciting.
Yeah. So data sources, I have about 5 minutes left, so I will go a little bit quick on these. Safety data is of the utmost importance. A lot of the things we're doing are with implanted devices. Some of this slide is talking about differences in the data sources. There's adverse event coding that can be different. There may be systematic differences in data collection. And part of the things that we're talking about now is data that may come off different clinical trials or different sources where it may be very high quality data. So we're not necessarily concerned with there being missing data, but it just may be fundamentally collected differently. What then we'll end up with is you have to expose that data into adjudication committees and get it harmonized in a way that makes it amenable to analyzing against a future data set. I talked about radiographic data. Again, most trials that we're doing require radiographic data. There's differences in modalities that you have to be on the lookout for. There's differences in who's doing the reads, where the reads are, do you have access to the source data? And a lot of the trials we're doing, things may be shifted. You may not be able to use a certain data source if you can't go back and actually get the physical images to actually do rereads on for any new trial. There's challenges in every domain of doing all these. I've alluded to all of these, so I will only pause there for 1 second. I'm going back to this slide and saying, well, what about next? This draft or this guidance document was already discussed earlier, and I'm pulling out 3 different components of this. So what's in this FDA guidance document is talking about, is the data fit for use, and can this meet particular regulatory considerations? What this document doesn't have, and I wouldn't expect it would be, is another 4th bullet, and that's going to be all about how you actually get that data, and that's part of our work and continues to be more and more central to what we're doing and where I think you all can be extremely helpful in. So I inserted this bullet in there, and I'm just calling it the other bullet because what this really represents is how do you get these data sources? We can see that FDA is progressively pushing and allowing for these type of trials. Where does this data come from? and that will be my last slide. But where this data can come from is a lot of different places. One may be competitors on the market. There may be previous trials that you have conducted. But what we're consistently brought in to do is to evaluate, can we use these data sources, and are they eventually going to be able to be poolable and be able to support a particular trial? This will also involve very, very large transactions for these data sources. So while I was saying that it may be not feasible to enroll a new study, there are cases when you may have a direct competitor and there may be only 2 or 3 in that particular space, but the only way to actually get the data is to go to a competitor. There are things you have to be on the lookout for when you're evaluating those type of data sources, but there is a lot of collaboration, but it will be through some external party. We do it. There's a couple of other companies that are also in that field working on, but that is purely what you all are trained to do, is to evaluate these disparate data sets and really truly understand, can these support the inferential scope that you're going after? So there's a lot of opportunities for us to continue to borrow the research that you are all doing. So please don't stop doing that, because I like being able to borrow all these great ideas. It is really, really helpful for us. We're absolutely reviewing all the different literature that you do, even as applied statisticians. So if anything, I hope you leave this talk by realizing that there is a lot of collaboration. There's a lot more similarities, and we've been borrowing your ideas for a while and plan to do so going forward. That's what I got. And should I take my questions in the panel?
Yep.
All right.
There's a fire warden for each wing that will tell you whether you need to evacuate or not. So she was outside. All clear.
Alrighty, so now it's time for questions. And we know everybody's awake. We know everybody is like ready to go because you've just had both an amazing statistical talk and the ability to stand up due to an alarm. So what are the questions for today?
I have a question for David. So I'm very familiar with IBTW. It's the first time I hear the terminology subclassification. I just want to make sure maybe I misunderstood from your slide, or maybe because I was typing, I missed something you said, but I got the impression from the slide that you showed us where you had your area of good overlap and then the outliers, that what you end up doing with subclassification is almost like a subgroup, like you restrict the analysis to the group with better overlap. Isn't that just the same thing as trimming, but not using the terminology trimming?
So what we're trying to do is we're trying to get
You're on. You just have to lean in.
so what we're trying to do is to get essentially 5 different randomized trials in one. The constraint that we have is that we can't do trimming on the investigational side, which I was talking about. When you have controls who are so far out and would not be there, you absolutely would trim them in the same kind of way. So you may end up with a very, very small weight in one world. In the other world, you may trim them to continue to produce balance that meets a pre-specified set of criteria.
And
Okay, I
if
missed that it was in the context of our CTs.
also, if you were to go to the propensity score pre-conference course here, they would call it stratification.
Oh, okay. That's what I got the impression. It was just different terminology. Okay, thank you.
You think about the big picture. Medicines are looking for adverse reactions to biomedicine based on the risk management plans, potential risks, so on. Yeah, the adverse reactions are mostly lack of effectiveness or defect, or are they adverse reactions in the sense that the medicines you can get hepatic or renal or whatever is good for them? So what are you looking when you're monitoring the safety of the device? Is it the malfunctioning, or is it causing emboli or something? This is one thing. Because that determines the comparator. Do you do the registry without a comparator? Is the comparator standard therapy as it is with medicine? Is there an existing device, or is the new device improving? I'm trying to get your head around what are you really looking for? Adverse events or lack of efficacy? Are you talking about comparative effectiveness or comparative safety?
I'll start, and Paul, I'm assuming you're going to jump in here. Just like drugs, we can do outcome studies for effectiveness and for safety. When we are looking at device outcomes in a safety realm, there are indeed patient-related outcomes. Those are the ones that are very much akin to what you would think about for a drug. We also, in addition to all of those that we would for drugs, have malfunctions, procedure-related events, facility-related events, and device-related events. All of those need to be part of a portfolio for safety for a device. And so it's everything for drugs and then some.
Thanks. Just to add to that, in like, say, in drugs, the safety events are considered adverse events, called adverse events. In devices, they're called complaints because they include adverse events, but often the complaints are, as you say, that the device doesn't operate well or it malfunctions, something goes wrong, because they're often mechanical devices, and so they don't often work. Sometimes they don't work well. So that complaint is a broad umbrella. And what's interesting, I found when I moved over from the pharmaceutical side to the medical device side, is that in the pharmaceutical side in companies, the adverse event reporting is in the safety group, because most of the reports, the post-marketing reports that are coming in are adverse effects. Sometimes they're that the drug doesn't work, but mostly they kind of safety issues or sometimes packaging issues with drugs, but mostly safety-related issues. Whereas in the medical device side, the adverse events are in the quality group. So the quality group does the adverse event reporting or complaint reporting, because most often they're complaints around the quality of the product. So it's quite different in that way.
If you don't ask us questions, then David has really hard questions for us. Yes.
Thank you very much for a lovely review presentation of sort of current state of play. Advice, we enjoyed it. I had a question. Sorry, it's Devon Brock from the DSIU. I had a question regarding when it comes to the iterations of a product. Obviously, you mentioned products having quite a short life cycle, and in that sense, if there's a sort of battery of safety work being conducted for an iteration, then how do the regulators view future iterations? And is there a sort of criteria for, if the product substantially changes, then you're going to have to start again? Or, if it's a subtle change, then that body of work will suffice?
Oh, everybody's going to have opinions on this because everybody's working in this area. From the regulatory perspective, if you want the in-depth version of this, the FDA has all of the 522 studies and all of the past studies online. And so you can look to see what the description is of that study and what the results are of that study. If one of those exists, you need to complete that study on that product. And so there are often the previous iteration of an implanted device that's no longer implanted on anyone and hasn't been for 5 years. We're still tracking those people to learn about what happens for that device. The second iteration that they've stopped using 3 years ago, they're still tracking those individuals who have it. And the one that is just coming onto the market now, they're going to continue to track that one as well. And so all of those things are happening. We're learning from all of those studies of the iterations together. It's just that the earlier versions, we are further along in that learning. And that's typically how it gets handled. It looks a little different for wearables, and I'm assuming you've had to deal a bit with that. And then
Yeah, I was going to comment that from like an on-field perspective for us, if I look at the knee brace study, right now we have 15 different knee braces from the same company that are essentially doing the same thing. And from our perspective, those iterations are meaningfully different, even though I think from an approval perspective, they're out there and they're being offered to players. But from an actual effectiveness perspective, it sort of puts us back to part one, where we need to test for our perspective, like whether that will actually reduce forces in our population. And so for us, it's challenging because there are so many iterations and little tweaks that happen over time that I don't think from a on-field effectiveness perspective, we can assume that the next iterations will be equally as effective without at least testing at the beginning. I think in terms of what we're doing big surveillance studies looking at injury rates. Where we would probably lean towards is not necessarily needing to do that whole process, but doing some sort of lab testing to see if the qualities are similar to what we have previously evaluated.
Thanks, David, for that question. So I think the technical term for that is substantial equivalence. So if you can use, is there a predicate device with a substantial equivalence? So the criteria for substantial equivalence, I haven't seen a very well structured algorithm on this, but it's essentially it's influenced by two factors. One is the risk of the device to the patient, to the life of the patient. So that goes into the class 1, 2, or 3 device. So if it's life-threatening, then it'll be class 1. If it's very low risk, like say a knee brace, where or toothbrush, it may cause some bleeding of the gum, but it's not going to kill the patient. So so that would be a class 3. So the equivalence criteria would depend on how risky this is to the patient. And then the second factor is whether it's a 510K pathway or a PMA. If it's a 510K pathway, it's typically considered a low-risk device where there's well-established equivalence, versus a PMA is like a high-risk device that's new. So if it's a PMA device, then the equivalence criteria will be much more scrutinized. If it's like a 510K, then it's considered what would be a 510K example would be, say, sutures. If you come up with a suture that has a bar, like a knot in it, a pre-tied knot, so so whether that the pre-tied knot saves the surgeon from tying that knot, so it speeds up the procedure a little bit. It's not a huge risk to the patient. You can expect that the knotted thing is going to work just as well as the original thing unless the knot unties. But if it does untie, then it's not the end of the world for the surgeon or the patient. It's just going to take a bit longer to fix that. So there the substantial equivalence criteria would be much more lax.
And I'll just add, so it's very, very dependent on what the device is and what the change is. There's some changes that you maybe would show from just the bench, and then at the same time, it's not consistent where how applicable this is to any given device, because in parts of the world, you may have that generation that's already approved and being used, and you may have data that's collected there. So it's really dependent on where it is in the life cycle of that product, where else it's approved, and whether you can do it from the bench, and then to get into kind of the things that Paul was getting at, whether it ends up as 510K. So I'm not able to directly answer your question because it's very, very specific to the particular product, but there are mechanisms to not have to redo one of these very, very large, expensive trials again, but it's very common that you do. So it's really dependent on the product. The answer is yes. There's absolutely differences across the branches. I'll look over to some of my friends over here to expand on that, but the answer is yes. There's very large differences depending on which branch you're in.
Yeah, cardiovascular is the most willing to accept real-world evidence. When you look at the label extensions, most of them have come through the cardiovascular division. And I think probably the least is orthopedics, even though So I think it depends on how you define real world evidence, because orthopedics, they use a lot of registries. Why? Because, as Mary Beth said, sometimes, if you get a hip replacement, you want that to last your lifetime. You don't want to have to go back after 5 years and get a whole new, pull that hip implant out and put a new one in. So there, the registries are able to really look at long-term follow-up. And so in OHT6, the orthopedic, they're used to looking at registries. But when it comes to electronic health record databases or hospital databases, they're much more reluctant to use that, in large part because they say that they really need the x-rays. That in orthopedic medicine, the x-rays and the radiographic imaging are critical to evaluate whether there's been good fusion, whether the device is really sitting well. And so they want the x-rays, and often you don't have that in the EHR or the hospital databases. But also I think, when you look at orthopedics, it's governed by surgeons figuring out, it's a very sophisticated carpentry. And so it's governed by surgeons kind of figuring out empirically what works. And they get a pretty good assessment. Surgeons who are doing a lot of surgeries get a good sense of what works and doesn't work. So you don't typically see in cardiovascular medicine, you see 10,000, 30,000 patient trials looking for large outcomes over 5-10 years. That's the field of cardiovascular medicine. Orthopedics, it's really traditionally been surgeons figuring out empirically how do they get a good fit, and they can get a pretty good sense of that shortly after the patient, when they do the x-ray or within 30 days. So they're getting that feedback all the time. Sometimes it's a question of long-term outcomes in terms of infections, but that's a rarer outcome. So the field of orthopedics has evolved differently from cardiovascular medicine. I think that influences the willingness to accept these large databases where you're kind of looking at incidence rates and the kind of tools that we use in epidemiology.
I think I'm gonna disagree in general.
Well, Mary Beth is has much more expertise than me.
No,
So.
I think we like to have clean buckets, and I think that there are definitely some eases in cardiovascular compared to other areas, some eases in diagnostics compared to other areas. I agree that the hurdle for orthopedics is very high. I don't think that the actual consideration of therapeutic area is the right one. I don't think that it's the therapeutic area that's driving it at all. I think it is the expertise of the individuals who are presenting information to regulators. When you have someone on your team who understands how to explain the real world data, like Paul or like David, it's a huge difference. Those teams have a better experience with FDA, with the EU EC, with China, with the PMDA. I think that it is being able to talk to an engineer, to a clinician, and to someone who has a lot on their plate and has just a few minutes for this.
And a biostatistician.
And a biostatistician, in the way that they can understand this information. That is the bar. And if you have a data source, and if you have an endpoint that you can justify meaningfully, and you can explain that to that team, those are the groups that are better suited for this. I don't think it's by therapeutic area.
Yeah, I would echo that, and going back on a little bit what I said, the place where we see that there may be some differences in the branches, it's also the differences in who's doing the review. There's different levels of maturity in the reviewers. You could swap branches, so it's not necessarily a branch effect, it's a reviewer effect.
Yeah, I was just gonna, I know you had a second question. I think you asked what's right before this, and I think orthopedics is. And I've worked in orthopedics now for 15 years, and I think it's just an exposure. They don't have a lot of exposure to epidemiology in general in their big research institutes. And so I think if we can get to the place where we can kind of advocate for our skill set, we can get to the place where we can use more real world evidence in that space. But I know you had a second question too.
Yeah, so my second question is more on the methodologic side of things. So, I've seen references often to performance goals, and I'm curious when I think about the performance
so open more in the medical device world. What is driving that? And is there a situation where that is more acceptable than what the traditional kind of effectiveness estimates? If that makes sense.
Yes, this is my answer to your first question that I was debating whether I should provide or not. The biggest hurdle that we have as a field to being able to speak into all that is going on in devices is that we haven't taken the time to learn the language. There's a lot of language differences between drugs and devices when you move to a regulator and you start talking to them. If you're going to be talking to a regulator, you need to know their language and you need to use their language. That's kind of what we do across the board for drugs. You have to do it in devices too. Performance goals, objective performance criteria, they are standard pieces of study designs. You use them when they're appropriate to you to use given the risk and the study design. It's not something that is, whether you're using real world evidence or whether you're using an RCT, it's just that kind of thing. And for us, given how we are trained, it's more of a feel of traditional surveillance, not pharmacoepi. And so once you kind of like can translate from us from pharmacoepi into, we have had to learn as part of our epidemiological training about how surveillance was done and what that looks like. And once you take all of that information and pull it back over to in this specific device area, they use an OPC or they use a performance goal, you do the same thing. And it has zero to do with whether you're using real world data.
I would add to that one of the things that's hard to do in medical devices is randomize, because of what Mary Beth talked about is the surgeon learning effect. So if you're going to get a new device versus the device that the surgeon's used for 20 years, if you randomize to use one or the other, the new device they're just not going to know how to use as well. So you can't really randomize surgeons to use one device or the other. Secondly, you can't blind it because it's very apparent. So, and thirdly, often, you don't really want to, in many situations with medical devices, you don't really want to randomize patients because the stakes are so high. And I'll show one example in my talk later. And fourthly, I think there's the budget for clinical studies is much lower for medical devices, and FDA has a mandate to kind of keep the cost low. And then fifthly, there's an issue of like medical devices. I use it like an analogy of like an iPhone. So from your iPhone 11 to iPhone 12 to 13 to 14, they're updated, but it's not like a whole new kind of device. So, staplers, how to staple a wound, that's been around for hundreds of years. And iteratively, we keep improving that, thank goodness, because it makes surgeries quicker and better and safer and more effective. So as you iterate to a new generation of staplers to when you do a surgery and you're stapling up the vessels that have been cut or the tissue that's been cut, you don't need to really have a control group for the old stapler, because there's been so many studies done with that old stapler that you have really good data. So why have a control arm that you can't randomize? It's going to cost a lot of money. It may not be fair to patients when you can just get that data from the past clinical trials and published studies and use that.
Registry, we have the registry, the device registry. For a device registry, do you sort of at the time of giving the device or inserting the device or whatever, the prescriber get the consent of the patient that they will be included in the registry, and then you could monitor them both using electronic data as well as information from them. I mean, what's the procedures for the registry, or are the registries done exclusively on secondary data sources like claims registries and so on? And also, do you use disease registries like Parkinson's disease for looking at people who get devices inserted for the treatment of Parkinson's disease? I would imagine that you use both. I'm interested in the product registry with the consent of patients. Is it primary data collection or is it hybrid of primary enrollment and secondary data sources?
I think all of the above. I think definitely for orthopedic devices, you can be registered into a prospective registry at the time that your device is implanted. For us, we do almost like a disease registry, like you're talking about, where we're collecting players for our athletes that have a specific condition and then looking to see what sort of treatments they get. And that's more secondary data sources with the EMR. So it could be all of the above.
I think there's a mix depending on the purpose of the registry. So often registries are done for quality control on the procedures because many physician societies or healthcare systems, there's a lot that can go wrong on the quality side in terms of the practice of medicine, infections, bleeds. So they're often doing tracking registries. Like the American Heart Association has a number of different registries that they track, so they can track quality and figure out based on institutions, which hospitals are performing well, which are not, which surgeons are performing well, which are not, so they can help kind of give feedback to improve. And then sometimes those are used for research purposes. We work with a registry at Mayo Clinic that's for cardiac arrhythmias, and that's all extracted from EHR. Joint registries, orthopedic registries, typically PROMS is a very key outcome because whether or not the patient can walk well and function well, there you need the patient-reported outcomes. So there you need the patient consent. But so it's a mixture depending on the purpose.
And then wound care registries, women's health registries, those also look very different from the ones that Paul was mentioning as well. All of these are available for use. If a data source can be used in order to identify the exposure, the appropriate covariates, and the outcome, it is fair game across the board for devices. And if you'd like to read a lot about this, the International Medical Device Regulators Forum, IMDRF, has an extensive document specifically on registries, and it covers that all of these things both can be used and are used today around the world.
Just another follow-up. In the registries, if there are disease registries, you can't be comparative. They're not ideal. People who don't use the device. There will be zillion differences, physical matches, so on. But other examples of registries who use, like for example, say the arrhythmias registries, people who use the device, and then you identify in the arrhythmia registries people who have the same arrhythmia but don't use the device. Obviously, there'll be reasons why they didn't use the device. Other comparative studies in the disease registries, like example studies.
This is why I mentioned women's health, because in women's health, they specifically do that. The American Urogynecological Society has a registry that covers women who have pelvic organ prolapse, women who have stress urinary incontinence across the board, and it's how they are treated for that condition. It does include all treatments. It's not everyone who has the condition, but it is not just the surgical. It's not just the device. It's all options. But many device registries are device specific.
Like the National Joint, probably one of the best orthopedic registries is the British National Joint Registry, which is run out of Oxford University, and the people who get in are people who have a joint replacement, and there's also a very good Australian joint registry. Again, the denominator is people who have a joint replacement. So there, you comparing against people who don't have a surgical procedure is difficult because they're not getting in. But in the women's health, then they are.
And then I'll add that there's a lot of work actually being done to prospectively set up these registries going forward. And so it may be that in one particular area, all patients who come in will be tracked regardless of whether they get the device. So it's right for that. And it's really, you're going to have the same level of data. Is it available today? No, but there's a lot of places where this is getting started right now to have those data sets to be able to do it. And that's a great data set. It's just everybody who's exposed to that physician or that practice is tracked regardless of the device or not. There's obviously a world of things you can do with those type of data sets, and they're actively ongoing. There's a lot of work being done in them.
And this is one of those skill sets that you in this audience can contribute to, not just next year, but over the next 20 years. Because Mary Beth said this is a hugely growing area, and the epidemiologists are really going to be needed to understand what data sources are available and how they can be used, so that when a research question, whether it's a safety or effectiveness question, comes up, you understand what data sources are available, how they can be used, what registries are available, where they can be used. If they can't use a registry, can you supplement, can you link to EHR data to get long-term outcomes, etc.
Any more questions?
I think we are at time. Mackenzie's like, is this time? Can we?
We're getting close.
I think we are very close. Is there a burning question from anybody?
I suppose you touched on it, I think, in your early talk, about sort of drug-device combinations. I suppose from a regulatory perspective, is that sort of handled by medicines?
Oh, this is where there's a lot of hand waving in combination products that are drug device combinations. So it looks a little bit different in different contexts. In the United States, there is a separate office that makes a determination on whether this is primarily a drug or primarily a device. For instance, a product that is a drug delivery with a device where the drug is already there, if that is outside the body, it is usually primarily a drug. If the device that houses the drug is implanted, it is usually primarily a device. In the United States, wherever this determination is made, that office covers the review. They will consult to the other group. And so if it's deemed primarily a device, then the device center will handle it. They will consult to the drug center, and then vice versa if it's deemed primarily a drug. However, in Europe, a drug device combination must undergo, if it is a drug delivery device, it must undergo all of the device regulation and then go to the EMA for the drug regulation.
I'll add just one brief thing on that. I see it from the sponsor side. Thinking about that determination, it can have fundamentally different meanings for how their next couple of years are going to play out. It's very, very, very different, all the way down to site operations. I mean, it's a fundamentally different study where that's made. So eventually, I think it would be great if we see a little bit more clarity and predictability in that, because it's not 100%.
There were some pretty major legal battles that played out over the past couple of years, and I've worked with a number of companies that if it is generally considered a device around the world, they often will argue it, they'll appeal it, rather than try to go down the drug realm if it's deemed a drug in the United States. It's just untenable.
Yeah.
Generally. All right, Paul, I think we are to you to help us finish out for the day. Hold on to your seat. Paul has really exciting, cool stuff that's hot off the presses.
Well, last but not least, hopefully, so firstly, thank you, Mary Beth, for organizing this course and for including me in it, and also thank you to our Canadian hosts in this beautiful part of the world. So I'm going to talk a little bit about. So just by way of background, so we've heard Mary Beth coming out of until recently the chief scientific officer or chief epidemiology officer for FDA, with a lot of consulting history. David as a biostatistician working for a consulting company, McKinsey, and Christina working for IQVIA, probably the largest kind of industry partner in overall. And so the perspective I'll provide is from being on the sponsor side and on the manufacturer side. So I work for Johnson & Johnson, which is the second biggest medical device company. The largest is Medtronic. We're a global company, as is Medtronic, and we have these different groups, like a pretty diverse portfolio of medical devices. So we cover a large group of medical devices. My experience in submitting applications to the orthopedic division versus the cardiovascular is there was a lot more resistance from the orthopedic division because they needed X-rays. And we just didn't have the X-rays, and it was just too complicated to get the X-rays. But if you could get the X-rays, which means that you had to go to a registry, because the registries generally have the X-rays, then you could get that. But I think that orthopedics is tremendous. There's tremendous opportunity for real-world evidence. So because there's so many questions that could be answered with real-world evidence from databases that are not being answered, and so which makes the whole field of medical device epidemiology really interesting. I worked because there's so much opportunity to make an impact, and that's for two reasons. Firstly, because there hasn't been a lot of work in medical device epidemiology. So as we bring our skills to the questions at hand, there's just a lot of opportunities for people to shape the field and lay the foundations and advance the field. And secondly, because when I was in medical school, they taught us there's two branches of medicine. There's surgery and there's medicine. And most of 99% of what this conference focuses on is medicine, the field of medicine. But when you look at what happens in practice in medicine, it's probably 50% surgery and 50% drugs. And so that whole other part of medicine, the surgery part, is really unstudied. And so that's why our skills and our data sources and in epidemiology can be hugely influential in improving outcomes for patients, improving outcomes for society, improving costs. How many of you know someone who's had a hip replacement in the last, or knee replacement, or had an abdominal surgery to remove cysts, or to have a stent put in, or had a stroke and had something put in, removed a stroke from their brain with an emergency procedure, or had a heart valve put in. Just everyone's getting these procedures, and they're really extending life. But we don't really have good outcomes on what using the tools that we have, what's improving those outcomes, and how do we create a learning healthcare system in the area of surgery and medical devices, so we keep iteratively improving things for patients, for healthcare systems, that they can be more efficient, and ultimately improving, also looking at sustainability and environmental outcomes. It's a whole new area, because as we look at climate change, in making sure that procedures are more efficient from a climate perspective, is also an emerging area. So I'll be talking from that perspective. I'm also adjunct faculty at the University of Pennsylvania School of Medicine, and a fellow of the society. But also I've worked a lot with NEST, the National Evaluation System for Health Technology. I'm on the governing committee, and also the co-chair of the Data Quality Framework work group. So I'll try to provide some of that perspective. So just by way of disclosure, the opinions expressed are those as an individual employee of Johnson & Johnson, and not those of Johnson & Johnson as a company. So just to lay a little bit of foundation, this is a great paper by John Concato and Jacqueline Corrigan-Curry in FDA. They're on the CEDAR side, but Jacqueline Corrigan-Curry was the head of real-world evidence. The FDA has a cross division group, cross-division group that works on real-world evidence, so they can coordinate what's happening on real-world evidence across the FDA. And they wrote this paper, and I think what they talk about that the way we look at real-world evidence versus clinical trials is often we see them as two polar opposites, but in fact there's this continuum of randomized clinical interventional studies and non-randomized non-interventional studies. And increasingly we see them overlapping, real-world evidence being used for how to do a RCT, where to enroll people, how to recruit people, and then on the far extent, is the observational studies. That we're talking about cohort studies, but there's a lot of gray area. For example, for performance goals, if we have a single-arm clinical trial with performance goals, can we get those from real-world evidence? And often we do. And so, particularly in the area of medical devices, this kind of intermediate area where we're using both clinical data and real-world evidence is a big part of the evidence that's being used to understand how devices are working. Just wanted to mention this study that was published earlier this year in JAMA by some ISPE members, Shirley Wang and Sebastian Schneeweiss and a bunch of other folks, where they emulated 32 RCTs with non-randomized database analyses. And also, this was Etienne study, since we have 3 people from Etienne in the audience. Etienne did a lot of this work. And what they found is that if you looked at so they looked 32 pairs where they had an RCT, and then they did an observational study to match the RCT. And what they found, they used 4 metrics of agreement. If we just look at the Pearson correlation coefficient, there was about a 0.82 correlation in terms of the hazard ratio agreement between the two. But if we look at those where there was closer emulation on the scale of Picot, patient intervention, comparator outcome, and time, that Pearson correlation went up to 0.93, where there was weaker emulation. In other words, you couldn't quite, in the real-world evidence, you couldn't quite get exactly the elements that were studied in the clinical trial. It went down to 0.53. I think the very important lesson from this, that I take away from this, is that that if you can get the real-world evidence to match the clinical studies, you're getting a 0.93 correlation in the outcomes. But the biggest factor that's affecting whether you can get agreement between the RCT and the real-world evidence is, can the real-world evidence match the clinical trial? And oftentimes, in working with regulators, and in terms of using real-world evidence for label extensions or approvals, the biggest factor is, can you really get the real-world evidence to capture what you would have used in the clinical trials? If you can, then you have generally the regulators are much more willing to accept the studies, and we see this really provides analytical support for that. So what are I just reiterate what Mary Beth and Christina and David pointed out earlier, is there are these differences in clinical trial, in the development pathway for medical devices and pharmaceuticals, and that influences the role that real-world evidence plays in, because the real-world evidence is basically coming in to fill the evidence gaps that you have. Because, of course, if you can use RCT, you always use RCT. And so in pharma bio, you have these big phase 1 to phase 3 periods where you're getting a lot of evidence in the clinical trials. For medical devices, you have a big preclinical phase, as David alluded to earlier, because oftentimes you can test if it's a stapler or a cutter, or you can test how that's working in preclinical studies. Often we use pig tissue because pig tissue is often similar to humans. And then you can take it into a feasibility study, which is a first-in-human study in humans, and get a good sense of, is it working in a small number of subjects where you're monitoring very carefully. But you can see the clinical safety study is often quite small. In fact, for a 510K pathway, you wouldn't even have any clinical data, except maybe in China. So the post-marketing phase becomes much more important. And so real-world evidence really plays a much bigger role. So some of the factors are many low-risk products would not have any clinical evidence. If a clinical trial evidence is used in a regulatory submission, it's usually a single-arm study and really randomized. Trials are conducted in the hospital rather than outpatient setting, so then that affects you really doing hospital trials. Patient populations in device trials are generally smaller, since subjects are mostly surgical patients, so it's harder to recruit them. The The surgical technique, the surgeon experience, and the hospital procedure volume influence the study outcome, so we have to account for those. Blinding of surgeons is hard to do. Technological innovation proceeds more rapidly for devices than pharmaceuticals. So curiously, pharmaceuticals and biologics, generally you're dealing with innovation that happened 14 years ago, because by the time you get from the, all the kind of testing into phase 1, phase 2, phase 3, the technology transfer, the product, by the time it gets to market, it's taken 12 to 14 years. So you have time to do that big phase 3. Whereas with medical devices, you could, just like an iPhone, every year you can improve the iPhone. Every 2 years you can improve the iPhone. So that technological innovation is occurring continually, and therefore this ability to wait for these long clinical trials where your control group is still going to be relevant at the end is very different. Just to touch on types of real-world data sources. So Mary Beth talked a lot about MAUDE. But the hospital databases tend to be much more important for medical devices because that's where the devices are being used. And then, of course, electronic health records, because you can get the specific device out of the electronic health records. Claims, which are the staple in pharma epi, are important, but you can't get the medical device out of claims. So if you have a procedure code or that you can use as a proxy for the device, claims work fantastically, but most of the time you don't. Then, of course, registries are very important. But the problem with registries is the loss to follow-up rate over time. So typically, in the registry, as we talked about, if you've got a hip replacement, you want that to last 20 years. So you want to know, does this new hip replacement, is it going to last for 20 years as well as the previous device did? But by the time 20 years rolls by, you may only have, in some of the registries, it depends on whether the registry is run by the government or by companies. But when companies try run orthopedic registries, often you'll have 70% dropout rate by 10 years. So you can't really answer the question. But if you can link it to a claims database or EHR database, like a Medicare database or a SEER, then you can you get the rich database, rich device data and procedure data from the registry data, and then you link it to the long-term outcomes that you can get from the Medicare claims. So that linking is really important. Then there are things like patient surveys, surgeon surveys, PROMs, patient preferences, the wearables, the sensors, the social media, that are all very important. Why? Because oftentimes when you're looking at a new, say, new hip or knee, one of the things is you want it to last without a replacement, but you also want the patient to be able to walk well in it, to be able to play sports, to go back to running so they can exercise, because exercise predicts their long-term health outcomes from cardiovascular. So that you can only get from patient-reported outcomes. And Mackenzie knows way more about that than me. And then there's the device-generated data, because more and more the devices are digital and robotic. And so all of the time the device and these incredibly sophisticated navigation devices, just as you can use Google Maps, or I use Google Maps, you can use Waze or Apple Maps to drive your car around and get to places that you would have got lost otherwise. Well, that's the same thing that's happening in surgery. Surgeons can use these incredible navigation devices to figure out where to go in the surgery. Not only is it where to go, but also how to plan it. Because if you're doing a back surgery and you have to put 32 screws into the vertebra, and if one of those screws goes wrong, you either put the screw into the spinal cord and the patient can have go paralyzed, or they could have severe pain for the rest of their life, or you could break the vertebra and then the patient has to come back for another surgery, and it's incredibly painful and traumatic and disabling for the patient. So if you're going to put 32 screws in, if you have digital technology that will help you put the where to plan in advance where you're going to put the screws and what angles, and it'll help you to say, don't put it in on that angle because that's going to get too close to the wing of the vertebra. Put it more here because you're going to get the vertebra is not going to crack. Times 32, that's incredibly helpful for the surgeon. It speeds things up for the surgeon, and it gives much better long-term outcomes. But all of that device-generated data now on the back end becomes incredibly useful for people like you in the future to start to connect that to electronic health records and start to work out empirically what's working well for patients. So that's a novel data source for medical devices that you just don't get for pharma. So just touching on device identification, since that's such an important part of a medical device is sometimes you have procedure codes for if you're just looking at whether, say, a procedure like a hip replacement, you don't care whether it's manufacturer X or model Y that was implanted, but you're just trying to look at, like, do people got a hip replacement do better than people who didn't get a hip replacement? There, you can use the procedure code. You could use a claims database or any database, because all databases will have the procedure code. But if you want to look at a specific medical device, you're not going to get that from a claims. Claims don't contain specific medical devices unless the procedure code is synonymous with a specific device. We had a case with, say, a left atrial appendage. So one of the things that's used for people who have atrial fibrillation is you can close off the left atrial appendage, which is this little part of the heart where, because if you have atrial fibrillation, you can get a clot in that left atrial appendage, and if you close it off, it can prevent the patient from getting a stroke. Well, there's only one manufacturer who makes that device currently. So the procedure code is the same as the device. So therefore, you can use the procedure code. You could do a claims database study, because the the procedure code is equivalent to the medical device. But otherwise, you really have to go to these databases where you can pick up the specific device. Hospital billing statement often becomes a proxy for where you can get that device information, or NLP, natural language processing, of clinical records is where you can extract the data. So you have to do this kind of dance to get the device data out that you don't normally have to do in the pharma side. And that's why we haven't seen the same expansion of epidemiology in pharma, because in pharma there's a national drug NDC code, tells you exactly what drug what is used, what dosage, what manufacturer, and it's very simple. So the databases tend to be different, but you can use all. But often the hospital database provide a lot more information. The claims are very important. For cancer, Medicare SEER, but Medicare's are very important. And of course, the registries and the electronic health record systems of specific healthcare systems where you can extract the data. And then there's all these new emerging databases, such as Datavant, which aggregates data from across different integrated delivery networks. Datavant is Loopak does. Datavant is a group that does they create tokens that allow you to link. So they can be very helpful for linking between a registry and a claims database. And then various others. I won't go into all of them, but they all have their role in a particular way that can be used. And then, of course, outside the US, because oftentimes devices will be approved first outside of the US, and you could get you're trying to get information that's relevant for the Japanese situation or the Korean, what's relevant to Asian or European devices. So I think Mary Beth touched on this, but this is a great resource. I just want to reiterate it, which is this FDA document. The link to it is at the bottom, but if you just look, it's easy to find it on the FDA website, which lists out the 90 examples of real-world evidence being used for regulatory decisions for medical devices. It goes up to 2019, so anything since 2019 is not included, but it's really worth looking through to get a sense of the flavor. So most of the 82 out of the 90 are for therapeutic devices rather than for diagnostic. I think there was an earlier question about which areas do we see most use of medical device information for regulatory decisions, and this is a great document for this. I think 60% of the examples in this are for cardiovascular medicine. So, and I think there are 6 out of 90 that are orthopedic, but all of those are registries. So it gives you a good sense of where it's being used. And then there's also, I mentioned earlier, there's this pre-market approval, which is for devices that are higher risk and newer. And we've seen very little use for pre-market approval of using of electronic health record databases or claims databases. Most of the pre-market approvals have been kind of complementing registry data or clinical trial data. And I'll show two examples. So I'll show two examples that come out of this document that we just kind of selected out because they were kind of most relevant and often cited. So the first one is for a ventricular assist system device. This is the indication for use is as a bridge to cardiac transplantation in patients who are at risk of death from refractory end-stage left ventricular heart failure. So as you know, heart failure is basically, it's like cancer. It's a death sentence. It's a slow chronic disease. Very few patients recover unless you have a heart transplant, but often take a while to get a heart transplant. So this device was developed. It was indicated as a bridge to cardiac transplantation. I think more and more there's looking at whether you can actually start to get artificial pumps that'll replace the people who have heart failure, which is basically the heart, just not being able to pump anymore. But for these early stages, it was just really a bridge to transplantation. So the study design was that a non-randomized, open-label, prospective, contemporaneous control pivotal clinical study, and then the primary data was from this advanced trial with a treatment cohort of 140 patients implanted with this ventricular system device between 2008 and 2010. And the real-world evidence was used as a contemporaneous control for the clinical trial in roughly 500 patients that came from a registry, the INTERMACS registry, which is an interagency registry for mechanically assisted circulatory support, taken at the same time. Why is the time important? Anyone want to venture a guess? Yeah, and also that the practice of medicine really improves. So, constantly the physicians are figuring out what goes wrong and how to improve it. So if you come in if you have control group is 3 or 4 years later, the rate just may have improved tremendously. And what they found, so they used propensity score analysis, evaluated the comparability between the trial and the registry patient characteristics. So one of the things you see here is propensity scores are being used extensively in medical devices. It's really a bedrock of the use of real-world evidence here, just to make sure that the registry and the clinical trial are similar. We see, typically in pharma, there's a lot of propensity score work going on where you have two cohorts in the same database. Often in medical devices, it's been used between two different data sources to make the two different data sources comparable. And David, you mentioned that. So the test was a non-inferiority test with pre-specified non-inferiority margin. So why would it be non-inferiority test? Anyone want to venture a guess? Yeah. Also because because the goal here was not necessarily that it's better, as long as it's as good as the current standard of care. That's enough, because then in practice of medicine you have two options. So what they found was that this new device was not inferior to devices used in the control group in effectiveness. The overall safety profile was deemed acceptable for this high-risk patient population, but it was associated with higher neurological event rates, which is not surprising because the heart and the brain. We know we often have vascular dementia. People don't get enough blood supply to the brain, but the device was approved based on its non-inferiority. So this was using real-world evidence as a control group. Here's another example. This is probably the most famous example because here the real-world evidence was the treatment group, and the control group was a clinical trial. This is for people. The indication of interest is a transcatheter aortic valve. So why is it called transcatheter? Because previously, if people had heart valve problems, you had to do open heart surgery. Crack open the chest, huge, take hugely invasive surgery to repair the heart valve. What they figured out is you can go through the vein in the wrist, or the femoral vein, into the heart, and as the picture shows, you can basically cut out the old valve and put in a new valve. And they're different sorts. Some are PIG, some are synthetic, some have drug on them to prevent clotting, others don't. But the basic premise is this going through the trans catheter, rather than through open surgery. Now, it was initially approved for people at high risk. So high risk of death or complications of surgery. Basically these are people who, their heart valve starting to fail and they can't really have the surgery. It's basically like a death sentence. So if you can go through the aorta, you can now start to do much less invasive surgery and get much better outcomes. So who would be at high risk? People who are very frail, maybe have bleeding conditions. So but now the question was, could you use it for intermediate risks? And so they did the study where so they got good 30-day clinical outcomes in patients of intermediate risk of surgical mortality in the clinical trials. But they needed, for approval, the FDA said you needed longer-term outcome data for the label extension for the intermediate risk patients. So they created a real-world evidence. They got it from a registry. The registry. This registry was set up by the Vascular Society of Medicine, and it was largely set up because, partly just to look at quality of outcomes. So what were the outcomes to patients, but by the Society of Vascular Surgeons, but also because the hospitals participate in this because they needed to get the data for reimbursement, because for Medicare they had to get reimbursement. And so there was very high engagement by the people who were doing this procedure in the registry. So they had just under 1,000 patients in the registry who they could follow for longer term outcomes. And then the external control was this clinical trial where patients were treated for surgical valve replacement, and not too far back in 2011 to 13 versus the registry data from 2014. So that was with the surgical valve replacement for intermediate risk patients, so they could get that data from the control group from the clinical trial. And the primary endpoint was all-cause mortality and incidence of stroke through intervention in aortic valve regurgitation at 1 year, propensity scores to balance between differences between the clinical trial and the registry, and they looked at both a non-inferiority and a superiority analysis. And TAVR was both significantly non-inferior and superior to the surgical valve replacement for the primary composite endpoint. So this was approved. This led to label extension. It was published in Lancet. But one of the big things that why this was approved was that I think the effect size was like 70% reduction in death. So it's kind of like a no-brainer, So because one of the things that's not apparent from this FDA document is what's the effect size. Because if the effect size is really huge, particularly for a life-threatening outcome, you're much more willing to tolerate the uncertainty of real-world evidence. In situations where the effect size is much smaller, or for less, the threshold for using real-world evidence becomes a lot higher. Now, this is another example from China. This hasn't been published. This was a J&J example. But one of the things that so China really looked at what was happening with the FDA and the European, what in the European environment, and they sat back about it. Because I went over 3 times to China to meet with the Chinese NMPA, as did people from other companies, and IQVIA and others. And they had these consultations, 2 days or 1 day, where they asked all of us to present what we're doing with real world evidence and what was FDA doing and how were the companies responding to what FDA was doing. And you could see they're thinking about it and discussing it. And we were supposed to, one day we met in Beijing, we were supposed to finish at 5 o'clock. And they kept asking questions till 6 o'clock. So we stayed until 6 o'clock until the hotel kicked us out and we had to leave, which indicated they were really very much engaged in it. And then they came out with these guidance documents for how to use real world evidence in China, as it happened in Korea and other countries. And what they came up with was this idea that in the south of China is this province called Hainan. It's an island. And it's pretty close to Taiwan. And it's they created this as a special zone where before China's had this requirement that you have to do a clinical study of a device in China. So typically what would happen is you get things done through Europe and the US and other places, and then you'd have to do the clinical trial in China, and it would take 4 years by the time you got the clinical trial, enrolled all the patients, and got it on the market in China. So all the newest technology was not available in China. China has 5,000 medical device companies, so they also were they recognized strategically they were impairing their medical device industry to be competitive because they just didn't have an opportunity to test out the new technology. So they changed, and also many patients in China were going to Singapore or other places, Germany, for surgery because they could get the newer technology there. So what they created this special zone where you could use the newest technology if it was for life-threatening events and or important medical breakthroughs, and generate real-world evidence in this large hospital zone and use that real-world evidence to get approved in China. So instead of doing the clinical trial, you could just introduce the new device in this special zone. The Chinese patients could get access to the latest technology in China without having to go outside, and then they would generate the real-world evidence and use that instead of the clinical trial requirement, which is a very smart idea. So this is one technology that got approved through that pathway. It's for this Catalyst precision laser system, which is used for cataract surgery. And what it does is it uses imaging and guided delivery to generate accurate laser incisions for cataract surgery. As you can imagine, if you're putting a new lens in, cataract surgery is one of the most common surgeries in the world. Actually, just one of those things we take for granted, but really so many people would have been losing eyesight in older age now can continue to have great vision. But the accuracy which you make the cuts in the eye are really important, and if you can do that through laser technology, which with much greater precision, that really helps. So the Catalyst system was approved first in the EU, to aside earlier point, first in EU in 2011, and then subsequently in the USA in 2012, and it was approved in 64 other countries. But China would not approve it because they wanted that clinical data in China. And so finally it was approved in January 2021 through this special pathway where the robot was introduced in this Boer hospital in Hainan province. We generated real-world evidence, and then we used the real-world evidence to supplement the evidence on the worldwide clinical trials, clinical literature, pharmacovigilance evidence, because there's a lot of pharmacovigilance evidence, to address ethnic differences. So it's one really interesting example of how real-world evidence is being used. I wanted to turn to NEST, which is the National Evaluation System for Health Technology. So it was created by FDA as a public-private partnership, funded by FDA initially with seed funding, but also getting some funding from industry through the MEDUFA, which is the equivalent of PDUFA. And it's designed to foster the use of real-world evidence to develop new and safe technologies for medical devices. They created a research methods framework and a data quality framework, The methods framework is to guide the use of the development of methods for medical devices, and then the data quality to guide the criteria for data quality that can be used. The two frameworks were already published by NEST, but we're working on version 2.0 to try and come up with more specific and useful versions of these. NEST also has a whole bunch of collaborators that are part of through which studies can be done. And these are all collaborators in the US who've kind of agreed to have their data used through NEST if they elect to be part of different studies. This is some of the work on the data quality framework that I co-chair. There's a chapter on data governance, database source, extraction, transformation, and loading, all the quality procedures for that. Then once you've gotten the data source and you've extracted the data, what's the data quality for that particular study? Any work on linkage, and Mary Beth was the one who said we need to add a chapter on linkage when she was at the FDA. And then lastly, on the NEST data maturity model, which is a way of qualifying. One of the things that NEST is trying to do is kind of pre-qualify databases to say this database has gone through this quality control, and it has this, kind of like a Michelin 3-star, it's a good database. So that way it saves time from everyone having to redo it every time they want to do a study. So one of the criteria for data quality is the endpoint validation. Mostly identifying whether ICD-10 diagnostic and procedural codes and CPT codes accurately identify the safety and effectiveness outcomes. I won't go into a lot of detail on this. This is some of the work that we had to do. Covariate balancing. So one of the interesting things in the covariate balancing is the FDA statisticians have come up with this 2-stage outcome-free design to emulate randomization. And I think it's a really interesting approach by FDA. In many ways, Amy Abernathy gave a talk recently, and she said how these FDA statisticians are much more forward-thinking than what's happened on the pharma side, which I 100% agree with, because the clarity that this 2-stage design gives really allows a lot of innovation to occur. So the basic idea of the 2-stage design, which is not really a 2-stage design, it's really a 3-stage design, but anyway. So the first stage is external data source selected, clinical protocol development, sample size estimated, baseline covariance specified, independent statistician identified with no access to clinical outcomes data. So all the groundwork. The second step is the covariate balancing by an independent statistician who's blinded to the outcomes. So all the statistician is doing is they don't have any access to the outcomes. They're just doing the balancing. And they iteratively do that until the balance is achieved. Once they do that, they finalize a statistical analysis plan for future outcome. So the advantage of that is then you have this intermediate phase where you can really focus on balancing the cohort. And then the last stage is where you get the statistical inference of study parameters. This is done by a different statistician from the statistician doing the balancing, and you do the hypothesis testing analysis of covariate balanced data. So then you have a couple of different propensity score approaches you can use. There's propensity score matching, stratification, weighting, and that the inverse probability weighting or average treatment effect amongst the treated. There's something called stable balancing weighting, which is integer-based methods to give more exact matching and lose less sample size. And there are other methods. Just touch on stable balance weighting very quickly. We have a presentation on Sunday afternoon on this. We think it's a much better method than propensity score matching because propensity score matching uses least square error methods from regression. But what's happened in logistics and supply chain and operations research is there have been much more precise methods of matching that use more heavy-duty computer processing that we didn't have before, and they can give you much better matching. Stable. And the other thing about stable balance weighting is you can vary your matching criteria for different variables. With propensity score matching, you have to say, caliper 0.1. But it may be that some variables you really want to match very intensively because you can. Others you don't want, because if you match too intensively on that, you don't have a good match and you exclude a lot of patients. So this gives you that flexibility. So adjustment for surgeon effects are very important. Issue with surgeon effect is that often surgeons use one device or another. So you don't really have that equipoise at a surgeon level. A big area for new methods. If anyone's interested in methods in this field, figuring how to adjust for surgeon is a big factor. This is we just got this a label extension approved by FDA based on comparative real-world evidence study using EHR databases. Was from a NEST test case. I don't have time to go into it. Sorry, but it is published in JAMA and JAMIA. But we worked with Mayo Clinic, Mercy, and Yale. We did use the EHR database. It was a federated system, and we compared two devices and propensity score balancing, and went through. We describe a lot of what we went through, and we were able to show that there was non-inferiority, and got a label based on that. Here's an example of orthopedic example. We're looking at 2-year revision rates, where we can show a new hip replacement system works better than an older one. So I would say that I think looking at race and gender differences for medical devices is another big area for research. Very open. You can do it quite easily with databases, and something that hasn't been really studied. Another example is looking at using devices versus drugs. This is one example, looking at dementia risk for people who are treated with atrial fibrillation with ablation versus medications. And you can see that the people who got the medical device treatment, which is the blue dotted line, had a lower, a 41% lower dementia risk over the next 3 years. Cumulatively over 15 years, the people who got the cardiac ablation had a risk of 16% of dementia compared to 27% in antiarrhythmic drugs. The propensity score match. So you can start to look at drug versus devices. You can look versus early versus late treatment with medical devices. And here you have to be very careful of immortal time bias. So I'm going to the session this afternoon on immortal time bias, but that's a very interesting kind of look at would you treat early versus late with medical devices. And new technology, does new technology improve outcomes? This is a particular example. We can see the complications were 50% lower when the better imaging was used, and we just got a label extension by FDA for using ultrasound rather than X-ray based on this. This is just an example of some of the data that's collected. I mentioned some of the navigation data, or digital data. So this is a data source that maps the heart for atrial fibrillation. It uses CT, X-ray, and ultrasound, combines it into a combined data source, and we just were able to show better outcomes depending on that. So in conclusion, NEST has been great. We recommend you get involved with NEST. It's still going to continue. Real-world evidence is enormously informative. How to interpret real-world evidence uncertainty is really important and will need further development. Thank you.
Great. I will wrap us up in the last 2 minutes and encourage you to look through the examples that Paul just went through, because there's a lot of great information there. Let's see if I can get these slides up and fast enough. I just want to wrap us up by a couple of announcements for the rest of ISPE. I stole a couple of Mary Beth's slides, if I can get through my old slides quickly. I think we're almost there. To remind you that there's a lot of similarities in terms of how devices are studied to how drugs are studied. She had this great slide kind of showing that the life cycle is very similar. The timeline is very different, and the size of the studies and the type of the studies are very different. at the end of the day, our epidemiological training applies to devices too. So a lot of the same things that we're considering, confounding by indication, channeling bias, all of that is really the same in terms of how we study medical devices. But there are some unique differences, like provider characteristics. Paul just talked about, learning curve, and that we do need more methods development in terms of how we study the provider effect. And real world evidence and epidemiology really is key to how we study this total product life cycle. So please get involved. With that said, a couple of things to announce for the rest of ISPE. There's going to be a great session on Saturday that's hosted by the Medical Device SIG. There's a spotlight poster session that will be tomorrow on medical devices. You can check that out in your program. And then if you're interested in joining us for the SIG meeting, this is not in the program, unfortunately, but there is going to be a SIG meeting during the Friday evening SIG session. Just meet us at the registration desk and we'll find a place to go. So 7 p.m. we hope to see you there. And then there's reading. You can see this in the slides, but final reminder is just please do your course evaluation. It really helps us figure out how to modify this going forward. So when you get that email after the conference, don't delete it. Please fill it out. We appreciate it. And then we'll stick around. And if you have any other questions or comments for us, we're happy to chat more. But thank you all so much for attending, and please complete your evaluation.